Hello students, welcome to Vedantu Karnataka. Hegidira ellaru chennagidira. So namaskara for all of you. So students, first one hurdle niಮಗೆ cross ಮಾಡಬೇಕಂದ್ರೆ ಕಾಲೇಜ್ ಅಲ್ಲಿ that is going to be your FA1 ಇಲ್ಲಾಂದ್ರೆ first unit test ಅಲ್ವಾ. So first unit test ತುಂಬಾ ಕಷ್ಟ ಇರುತ್ತೆ because you would have just joined college, friends and all, you will be trying to go out and all that. So Vedantu Karnataka is going to help you for first unit test, which is not done anywhere else. It is just to make sure that throughout this journey, starting from your first unit test till your final exam or even supplementary exam, we were there. So we want to continue that this year also. Last year we joined in the middle, okay. So ನಾವು ತುಂಬ PU ಬೋರ್ಡ್ ಎಕ್ಸಾಮ್ ಮುಂಚೆ ನಿಮ್ಮ ಕ್ಲಾಸ್ ಎಲ್ಲ one shot ಎಲ್ಲ ಮಾಡಿದ್ದೀವಿ. But now we thought we will prepare you from your first unit test, and this is going to be the first chapter of your class 12th, right? Most of the colleges first 3 chapters irutte, kela colleges alli first 2 chapters will be there. So depending on what is your top, what is your syllabus, what is your thing, you should be focused, okay? So sexual reproduction, flowering plants, I'll be teaching one shot. So this is going to be a short lecture. This is mainly for all the unit tests. If you do not know anything about the chapter, watch this lecture. After that, solve the questions that I'm giving you. So these 2 things are more than enough for you to write your unit test really well. And students, unit test indane practice ಮಾಡಿದ್ರೆ that I want to score this much, or let's say I want to get at least 30 marks, and that if you consistently do, then your PUC exam is going to be very easy. Unit test full ignore ಮಾಡಿ, second unit test ignore ಮಾಡಿ, mid term ignore ಮಾಡಿ, final exam ಎಲ್ಲ ನಾನು ಮಾಡ್ತೀನಿ ಅಂದರೆ ತುಂಬ ಕಷ್ಟ ಆಗುತ್ತೆ students. So that's why it's always better you start from 0 so that it becomes faster. Clear? So sexual reproduction, flowering plants is the first chapter, and we will do an entire chapter one shot, okay? So what is this sexual reproduction? Yak sexual reproduction tumba important agide, so that variation can happen. Variation andre in students, now when you look, let's say when you go to someone's house, they just had a baby, you look at the baby and what will you tell? You look at the baby and you will be like, oh, baby looks exactly like the mother or the father, okay? Athara on prediction ಎಲ್ಲಿಂದ ಬಂತು? because of something called as your variation, that is the mothers and fathers characteristics will be there together for the baby, or mother's character will be more on the baby. So all that comes with your DNA and inheritance, right? So sexual reproduction in plants andre plants go athara on variation necessary, so they will be using male and female gamete to do the variation, okay? Yes, so look here, variation. I hope all of you are liking the video. Smash the like button, okay? So in angiosperm, students, all flowering plants will show sexual reproduction because flowers are the one that are going to bear the male gamete and the female gamete, okay? So sexual reproduction, formation of gamete and fertilization. So this is what the entire chapter talks about. So you'll notice all flowering plants show sexual reproduction, okay? This is important. All flowering plants will show sexual reproduction. Why? Because flowers are the main people who are going to have male and female gamete in them, okay? Now idella niಮಗೆ ಅಷ್ಟು ಇಂಪಾರ್ಟೆಂಟ್ ಇಲ್ಲ. Now how flowers are formed, okay? Flowers are, so NCERT snippu ಹಾಕಿದ್ದೀನಿ, so that wherever possible ನಾನು NCERT highlight ಮಾಡಿದ್ದೀನಿ, so that also you can go through. ನಿಮ್ಮ NCERT ಯಲ್ಲೂ ಆ ಥರ highlight ಮಾಡ್ಬೋದು, ಇಲ್ಲಾಂದರೆ ಈ ನೋಟ್ಸ್ ನಿಮಗೆ ವೇದಾಂತು ಕರ್ನಾಟಕ ಟೆಲಿಗ್ರಾಮ್ ಚಾನಲಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಅದು ಅಲ್ಲಿಂದ ಡೌನ್ಲೋಡ್ ಮಾಡ್ಬೋದು. ಟೆಲಿಗ್ರಾಮ್ ಇಲ್ಲ ಅಂದರೆ ಡಿಸ್ಕ್ರಿಪ್ಷನ್ ಬಾಕ್ಸಲ್ಲೂ ಇದು PDF ಲಿಂಕ್ ಇರುತ್ತೆ, ಓಕೆ? So look here, from the shoot, students, on the shoot there is going to be apical meristem. Meristem cells andre eno? The cells which are actively dividing. They are always ready to divide, okay? They are never going to stop dividing, so they are actively dividing cells. Our cells ige ಸ್ವಲ್ಪ plant already decide ಮಾಡಿರುತ್ತೆ, okay, I want to bear flowers, okay, I want to form flowers. Now ivaga if all of you are in this class, okay, be it Ganesh, Sakshi, Jyoti, Sunita, Uday, all of you are in this class because of a reason, that okay, I want to attend or I want to score well in my first unit test. That means ನೀವು already mind ಅಲ್ಲಿ prepare ಮಾಡಿದೀರಾ that I want to score this much marks in my first unit test, right? So like that only, even the flower also has already decided that the plant has decided that I want to bear a flower. So adik needed, like whatever is needed for that, be it the structural or hormonal changes, plant already start ಮಾಡಿದ್ದಾರೆ, okay? So here, look here, see, even plants go through preparatory phase, not just you, okay? Plants also go through this preparatory phase. So SAM, so here you see the shoot apical meristems, there will be internal changes. What are the internal cues? That is hormonal changes will be there, leading to the formation of floral meristem. Floral meristem in the directly nam flower formation agutte, okay. Floral meristems are going to differentiate and give us the entire flower. Now, it's very important for you to know the flower structure. Now, flower structure, students, most important, flower alli 4 whorls are there, okay. What is there? 4 whorls are there. 4 whorls andre starting with calyx, which is going to have the sepal. Nodi illi sepal nodidira most of you, neevu flower bud stage alli, okay. Bud stage alli time alli what will happen? There will be sepals covering them for protection, okay. So here the calyx is going to be sepal, corolla is going to be petal, androecium, stamen, gynoecium, pistil and carpel. That means what? Calyx whorl will have many units of sepals, okay. Corolla whorl will have many units of petals. Androecium whorl will have many units of stamen. Gynoecium whorl will have many units of pistil and carpel, okay. Understood? Yes. Now, here, look here, this entire flower, okay, this entire flower, what and all can you see? You'll notice, students, first you know stigma. Stigma andre pollen grains land aga on portion adu, okay. So, pollen grains are going to land on the stigma. So, stigma is right here where the pollen grains are going to land, and then you have anther. Anther is the one that is going to have the pollen grains. Inside that, pollen grains are going to be there, okay. Then you see here, can you see a structure which is called your filament, okay. El madidini labeling, ill nodi filament, okay. Adamele ill nodi stigma style ovary ovules. Idella female part. Stigma style ovary and ovules are going to be the female part. Male part is going to be anther and filament. So, another blue will highlight madidini, pinkal ido female reproductive system highlight madidini, okay na. So, ill nodi this anther and filament together forms the stamen. Anther and filament together forms the stamen, okay. Now, look here, students, petals are there. Petals will come where? Corolla, okay. Sepals are there. Where is it going to come? In the calyx. And then down, you will see a swollen part which is called the thalamus or the receptacle, okay. Thalamus or the receptacle. And most of you would have given flowers to someone in your life, okay. You might have given flowers, you might not have given flowers. In case you have given flowers to someone, you will be holding the pedicel, okay. So, let's assume this is the flower, okay. Nan elli itkonta irodu adu pedicel, okay. In a flower that will be called the pedicel. Clear to all of you? Yes or no? So, this is the pedicel. So, now students, in this flower there is something called as essential and non-essential part. Essential part andre the ones that are going to directly help in reproduction. The ones that are going to directly help in reproduction is going to be the essential part, okay. Non-essential parts andre, okay, they are not directly involving in reproduction, but they will help us some or the other way. Essential parts yaar barutte? Androecium matte gynoecium barutte alva, because androecium bandbit male reproductive part, gynoecium bandbit female reproductive part. Non-essential illi yaar barutte, students? Corolla matte calyx, because petals idrene insects will get attracted, right. Calyx is there for protection. So, both are very, very important. Understood? Understood? So, this is your flower diagram. Now, what are the events that are happening in this entire chapter? In this entire chapter, the events that are happening. 1 second. Okay. Okay. What are the things that are happening in this entire chapter? The events that are happening is going to be pre-fertilization, fertilization, matte post-fertilization. Pre-fertilization andre enu students? So, on madve madak munche, avarondo pre-wedding shoot ಮಾಡ್ತಾರೆ ಅಲ್ವಾ. Pre-wedding shoot ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, they will be telling that save the date, this is the day that we are going to get married, ಆ ತರ photo shoots ಎಲ್ಲ ನಡೆಯುತ್ತೆ, okay. Photo shoots ಎಲ್ಲ ನಡೆಯುತ್ತೆ. Fertilization stage ಅಂದ್ರೆ ಏನು, where the actual wedding happens ಅಲ್ವಾ. ಅಂದ್ರೆ wedding happens ಅಂದ್ರೆ where, like, just imagine any event, ಒಂದು ಬರ್ತ್ಡೇ ಇರ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ ಒಂದು ಆನಿವರ್ಸರಿ ಇರ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ ಒಂದು ಮ್ಯಾರೇಜ್ ಇರ್ಬೋದು. ಸೊ ಒಂದು ಪ್ರೀ ಅಂದರೆ ಏನು, ನಾವು ಡ್ರೆಸ್ ಇಸ್ಕೊಳ್ಳೋ ಟೈಮು, ಟೈಲರಿಂಗ್ ಟೈಮು, ಮೇಕಪ್ ಎಲ್ಲ ಹಾಕೋದು, ಆ ಒಂದು ಟೈಮು ಪ್ರೀ. ಆ್ಯಕ್ಚುಲ್ ಈವೆಂಟ್ ನಡೆಯೋ ಟೈಮು, ಆ್ಯಕ್ಚುಲ್ ಈವೆಂಟ್ ನಡೆಯೋ ಟೈಮ್ ಏನು ಇಲ್ಲಿ ಫರ್ಟಿಲೈಸೇಷನ್. ಅದಾದಮೇಲೆ ಆಫ್ಟರ್ ದಟ್ ವಿಲ್ ಬಿ ವಿಸಿಟಿಂಗ್ ಈಚ್ ಅದರ್, ವಿ ವಿಲ್ ಬಿ ಲೆಟ್ಸ್ ಸೇ ವಿಲ್ ಬಿ ಪೋಸ್ಟಿಂಗ್ ಆಲ್ ದ ಫೋಟೋಸ್. ಸೊ ಲೈಕ್ ದ್ಯಾಟ್ ಓನ್ಲಿ ಹಿಯರ್ ಆಲ್ಸೋ. Pre-fertilization events andre before the fertilization happens, what are the things that we are going to prepare for? Okay, what are the things that we are going to prepare for? Yes, tell me, you tell me, Banu, where is all your friends? ನಿಮ್ಮ ಕಾಲೇಜಿನ ಎಲ್ಲ ಫ್ರೆಂಡ್ಸಿಗೂ ಶೇರ್ ಮಾಡಿ. ನೀವು ಶೇರ್ ಮಾಡಬೇಕು, ನಾನು ಎಲ್ಲಿಂದ ಶೇರ್ ಮಾಡೋದು? ನಿಮಗೆ ಕಾಲೇಜಲ್ಲಿ ಫ್ರೆಂಡ್ಸ್ ಇರುತ್ತಲ್ವಾ, ಅವ್ರ ಜೊತೆ ಶೇರ್ ಮಾಡಬೇಕು. ಎಲ್ಲರೂ ಫಸ್ಟ್ ಯೂನಿಟ್ ಟೆಸ್ಟ್ ಬರೀತಾರಲ್ವಾ, ಸೊ ಶೇರ್ ಇಟ್ ವಿತ್ ಆಲ್ ಯೋರ್ ಫ್ರೆಂಡ್ಸ್. ಆ ಥರ ಸ್ಮಾಲ್ ಥಿಂಗ್ಸ್ ಯು ಹ್ಯಾವ್ ಟು ಡೂ ಫಾರ್ ದ ಚಾನಲ್, ಓಕೆ, ಆ್ಯಂಡ್ ಫಾರ್ ಅಸ್. ಸೊ ನೋಡಿ ಇಲ್ಲಿ ಪ್ರೀ ಫರ್ಟಿಲೈಸೇಷನ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಫಾರ್ಮೇಷನ್ ಆಫ್ ದ ಗೆಮೀಟ್ಸ್. ಸೊ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗಬೇಕಂದ್ರೆ ದೇರ್ ಶುಡ್ ಬಿ ಪ್ರೆಸೆನ್ಸ್ ಆಫ್ ಗೆಮೀಟ್ಸ್ ಅಲ್ವಾ. ಸೊ ಆ ಗೆಮೀಟ್ ಫಾರ್ಮೇಷನ್ಗೆ ನಾವು ಪ್ರೀ ಫರ್ಟಿಲೈಸೇಷನ್ ಇವೆಂಟ್ ಅಂತ ಕರಿತೀವಿ. ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ಫಾರ್ಮೇಷನ್ ಆಫ್ ಗೆಮೀಟ್, ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಆ್ಯಂಡ್ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್. ಮೈಕ್ರೋಸ್ಪೋರೋಜೆನೆಸಿಸ್ ಆ್ಯಂಡ್ ಮೆಗಾಸ್ಪೋರೋಜೆನೆಸಿಸ್. ಫರ್ಟಿಲೈಸೇಷನ್ ಅಂದರೆ ಏನು? Actual meeting of gametes is going to happen. So that is going to happen with the help of pollination, neither by water, insect or wind. Okay. Post fertilization andre eno? Fertilization aadmele what are the things that is going to happen? Okay. Yes, seed matte fruit formation. So ultimately ಪ್ಲಾಂಟಿಗೆ ಒಂದು ಗೋಲ್ ಇದೆ. ಏನದು ಗೋಲು? ಐ ಹ್ಯಾವ್ ಟು ಫಾರ್ಮ್ ಸೀಡ್, ಐ ಹ್ಯಾವ್ ಟು ಫಾರ್ಮ್ ಫ್ರೂಟ್. ಅದಕ್ಕೆ ನಾನೊಂದು ಪ್ರೊಸೆಸ್ ಮಾಡಬೇಕಂದ್ರೆ ದಟ್ ವಿಲ್ ಬಿ ಯೋರ್ ಫರ್ಟಿಲೈಸೇಷನ್ ಆ್ಯಂಡ್ ಪ್ರೀ ಫರ್ಟಿಲೈಸೇಷನ್. ಇನ್ ಪ್ರೀ ಫರ್ಟಿಲೈಸೇಷನ್, ಟೂ ಮೋಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ಥಿಂಗ್ಸ್ that can come as a direct definition question, or they can ask you to explain that entire process, is going to be microsporogenesis and megasporogenesis. So ಇದು ಬಂದ್ಬಿಟ್ಟು ನಿಮ್ಮ FAಗೆ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್. ಓಕೆ okay, two very important. Clear? Coffee aitu. Okay, now look here, this is what is given in your NCERT, how the flower is formed. Nan already explained madidini. Several hormonal and structural changes happen leading to the floral primordium, leading to the formation of the entire flower. Okay, entire flower. Now coming to each of the portion, that is, first we will learn androecium, then we will study gynoecium. So as I told you, androecium means what? Your stamen is the one that is going to be there, right? So what is going to make the unit, students? Stamens, right? Stamen is going to make the unit. Stamen has what and all portion? Anther and filament, right? It has anther and filament. So this is the filament, this is the anther. ಇಲ್ಲಿ NCERT ಅಲ್ಲಿ ಒಂದು ಲೈನ್ ಮೆನ್ಷನ್ ಮಾಡಿದ್ದಾರೆ, ದಟ್ ಥಲಾಮಸ್. ಓಕೆ, ಸೊ ಲೆಟ್ಸ್ ಟೇಕ್ ಎಕ್ಸಾಂಪಲ್. ಓಕೆ, ಲೆಟ್ಸ್ ಟೇಕ್ ಎಕ್ಸಾಂಪಲ್. ನನ್ನ ಕೈಯಲ್ಲಿ ಈ ಕಪ್ ಇದೆ. ಓಕೆ, ಅಯ್ಯಯ್ಯೋ, ಓಕೆ ಕಪ್ ಬೇಡ. ಓಕೆ, ನನ್ನ ಕೈಯಲ್ಲಿ ಈ ಕಪ್ ಇದೆ. ಓಕೆ, ದಿಸ್ ಇಸ್ ವೆರಿ ಕ್ಲೋಸ್ ಟು ಮೈ ಫೇಸ್ ಅಲ್ವಾ. ಸೊ ಮ್ಯಾಮ್, ದಿಸ್ ಇಸ್ ಪ್ರಾಕ್ಸಿಮಲ್ ಟು ಯು. ಪ್ರಾಕ್ಸಿಮಲ್ ಅಂದರೆ ಏನು? ದಿಸ್ ಇಸ್ ವೆರಿ ಕ್ಲೋಸ್ ಟು ಮೀ. ಓಕೆ, ಲೆಟ್ಸ್ ಸೇ ಈ ಕಪ್ ಇವಾಗ ಇಲ್ಲಿದೆ. ಇದೇನು? ದಿಸ್ ಇಸ್ ಡಿಸ್ಟಲ್ ಎಂಡ್ ಟು ಮೀ. ದಟ್ ಮೀನ್ಸ್ ಇಟ್ಸ್ ವೆರಿ ಫಾರ್ ಫ್ರಮ್ ಮೀ ಅಲ್ವಾ. So let's say one small animal has to go, it has to go all the way like this. Correct? It's very far from me. Now if it is like this, it is very proximal to me. So thalamus ige, so near thalamus, just look here, to the thalamus, this portion. Okay, that means your filament is in the proximal end and anther is in the distal end. Okay, ನಾನು ಇದು part 1 part 2 ಮಾಡಿದ್ದೀನಿ, ಅದಲ್ಲಿ ಡೀಟೇಲ್ ಆಗಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಿದ್ದೀನಿ. ಬಟ್ ಇದು ಬಂದ್ಬಿಟ್ಟು ಒನ್ ಶಾರ್ಟ್, ಸೊ ನಾನು ಅಷ್ಟು ಡೀಟೇಲ್ ಆಗಿ ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡಲ್ಲ. ಬಟ್ ನಿಮಗೆ ಫಸ್ಟ್ ಯೂನಿಟ್ ಟೆಸ್ಟಿಗೆ ಬೇಕಿರೋ ಎಲ್ಲ ಕಂಟೆಂಟು ಎಕ್ಸ್ಪ್ಲೇನ್ ಮಾಡ್ತೀನಿ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ದಿಸ್ ಇಸ್ ವಾಟ್ ಇಸ್ ಇಟ್? ಲಾಂಗ್ ಸ್ಲೆಂಡರ್ ಸ್ಟ್ರಕ್ಚರ್ ಕಾಲ್ ದ ಫಿಲಮೆಂಟ್ ಆ್ಯಂಡ್ ದಿಸ್ ಬೈಲೋಬ್ಡ್ ಸ್ಟ್ರಕ್ಚರ್. ಬೈಲೋಬ್ಡ್ ಅಂದರೆ ಏನು ಮ್ಯಾಮ್? 2 ಲೋಬ್ಸ್ ಇದೆ. ಬೈಲೋಬ್ಡ್ ಅಂದರೆ ಏನು? 2 ಲೋಬ್ಸ್ ಅದೆ. ಓಕೆ, ಬೈಲೋಬ್ ಮೀನ್ಸ್ 2 ಲೋಬ್ಸ್ ಅದೆ. ಸೊ ಲೋಬ್ 1, ಲೋಬ್ 2. ನಾವು ಈ ಲೋಬ್, ಸಿಂಪಲ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ಲೋಬ್ ಯಾವ ಥರ ಇರುತ್ತೆ ಗೊತ್ತಾ? ಲೆಟ್ಸ್ ಅಸ್ಯೂಮ್ ಈ ಗ್ಲಾಸ್ ಇದೆ, ಇಲ್ಲೊಂದು ಗ್ಲಾಸ್ ಇದೆ, ಇಲ್ಲೊಂದು ಗ್ಲಾಸ್ ಇದೆ, ಇಲ್ಲೊಂದು ಗ್ಲಾಸ್ ಇದೆ. ಓಕೆ, ಐ ಡು ನಾಟ್ ಹ್ಯಾವ್ ದಟ್ ಮೆನಿ ಗ್ಲಾಸಸ್, ಐ ಶುಡ್ ಹ್ಯಾವ್ ಬಾಟ್ ಇಟ್. ಸೊ 1 2 3 4 ಗ್ಲಾಸಸ್ are there. 4 glasses are there means what is going to happen? Okay, here you see filament, long and slender, bilobe, 2 lobes are going to be there. Okay, ಇಲ್ಲಿ ನೋಡಿ, this lobe, no, if I cut right in the center, it is going to look like 2 cups here, 2 cups down. Correct? So if I cut this right in the center, it's going to look something like this. ಈ ಆಂಥರ್ ಒಳಗಡೆ ಏನಿರುತ್ತೆ ಸ್ಟೂಡೆಂಟ್ಸ್? ಆಂಥರ್ ಒಳಗಡೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇರುತ್ತೆ. ಓಕೆ, ಆಂಥರ್ ಒಳಗಡೆ ಏನಿರುತ್ತೆ? ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇರುತ್ತೆ. ಅಂಡರ್ಸ್ಟೂಡ್? ಕ್ಲಿಯರ್? ಓಕೆ, ನೌ ಮೂವಿಂಗ್ ಫಾರ್ವರ್ಡ್. ಸೊ ದಿಸ್ ಇಸ್ ವಾಟ್ ಇಸ್ ಯೋರ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಆಂಥರ್. ಓಕೆ, ದಿಸ್ ಇಸ್ ವಾಟ್ ಇಸ್ ಯೋರ್ ಸ್ಟ್ರಕ್ಚರ್ ಆಫ್ ಆಂಥರ್. ಸೊ ನಾನು ಆ ಕ್ರಾಸ್ ಸೆಕ್ಷನ್ ಎತ್ಬಿಟ್ಟು ಈ ಥರ, like let's say I flip it like this, how is it going to look? It is going to look like this. Now what are the most important things that you need to remember? Your anther is to protect the pollen grains. Okay, to protect the pollen grains formation, even the sporogenous tissue, they need to have multiple layers for protection. Correct alva? Our multiple layers yavdo? Epidermis, which is going to be the outermost layer which is lining the entire anther. Okay, then you have endothelium, which is the blue color one that all of you can see. Correct? blue color one, which is again a single outermost, next to the epidermis, okay? Adamele in students on multiple layers irutte. Okay, M for multi, M for middle layer anta remember madi. Middle layer where there are 1 to 3 layers, okay? I hope all of you are marking attendance. Come on, Uday, keep a track on attendance. Otherwise, these people will not mark their attendance. Come on, quickly. Okay, so look here. Epidermis, endothelium, middle layer, tapetum. So, epidermis bandbit outermost layer, adamele endothelium, adamele on multiple layers barute, which is the middle layer. So, nodi il bardidini 1 to 3 layers. Adamele students tapetum. Idalli 3 ellardu function explain madidare. Epidermis, endothelium, middle layer bandbitu helps in dehiscence and protection. Dehiscence andre eno, let's say nan kayal tumba chocolates ide, okay? Nanu kai open, like let's say I did not release it. Nanu open madilla andre nimage chocolate sikkutta ilva? Ma'am, nanage chocolate sikkalla ma'am. Same way. Let's say anther olagade pollen grains ide. If they don't open, pollen grains scatter agutta? Illa, pollen grains scatter agalla. Pollen grains scatter agilla andre it will not go and settle on the stigma. So then what's the point? What is the point? Plant is working so hard, so it can form a seed, fruit, it can do fertilization, but the anther is not ready to give the pollen grains. So adikon process ige now dehiscence anta heltivi. So let's assume anther ee tara ide. Okay, there's a line of dehiscence. Let's take one hand. There's a line of dehiscence. So that line of dehiscence will be there and all the pollen grains will get released. All the pollen grains will get released. Understood? So that will be called as, now all the pollen grains will get released. Now epidermis, endothelium and middle layer is going to do the job. Dehiscence and protection. Dehiscence and protection. Clear? Other students, tapetum. Tapetum is the innermost layer, but it has so much importance. It helps with nourishment. It helps with nourishment. Nourishment and ivaga now, let's say example, we put oil on our head, alva. Why do we do that? So that our hair gets nice nourishment. Oil olagade coconut irutte, olive oil irutte, castor oil irutte, almond oil irutte, ellaru different oil use ಮಾಡ್ತಾರೆ. Why do they do that? Because the hair should get the nourishment anta. ಆ ಥರ ಈ ಗ್ರೋಯಿಂಗ್ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ಗೆ ನರಿಶ್ಮೆಂಟ್ ಪ್ರೊವೈಡ್ ಮಾಡೋದು ಯಾರು? tapetum. Okay, tapetum is going to provide the nourishment. Clear? Tapetum is multinucleated, so you can remember binucleated also. Okay, binucleated also. Now listen here, students. In middle, I have drawn one blue color small cells alva. Adenado, that is called as the sporogenous tissue. This sporogenous tissue can potentially become the pollen mother cell. Mother illa andre entire process agutta? Illa ma'am agalla. So, a sporogenous tissue will get developed to the pollen mother cell, and that pollen mother cell is going to undergo microsporogenesis. Okay. So, look here. Now you have studied that the anther is bilobe. Now, students, this is one lobe, this is one lobe. Ee erad lobe na connect madadu eno? It's called a connective. What is it called? A connective. Okay. To attach both the lobes together, we have in middle what? We have a connective. Clear? Now look here. In one lobe, you have two thecas. Okay. This pink color which I have drawn. This is one theca. This is one theca. Not this circle. This space that they are providing. Okay. Two thecas are there. So we call the anther as dithecous. And now you see how many are there? 1, 2, 3, 4. These are your microsporangium. Okay. That entire thing is microsporangium. So we have 1, 2, 3, 4. Tetrasporangiate. Tetrasporangiate. Clear? Yes or no? Students, bega video na like ಮಾಡಿ. Okay? ತುಂಬಾ ಕಂಜೂಸ್ ಆಗಿ ಬರ್ತಾ ಇದ್ದೀರಾ ದಿನ. Very good. Okay. Yes. So, look here. Tetrasporangiate. So, 1, 2, 3, 4. So, we have 4 microsporangia. So, that is why we call it tetrasporangiate. Bilobe yake? Yerad lobe ide. Dithecous, why? Because in one lobe there are 2 thecas. Okay? And then tetrasporangiate because there are 4 microsporangia. Clear to all of you? And what are the 4 layers which are protecting them? Epidermis, endothelium, middle layer, tapetum. Elephant eats my tomato. Okay? Elephant, epidermis, eats endothelium, my middle layer, tapetum, tomato. Clear? Tapetum does the job of nourishment. Epidermis, endothelium and middle layer does the job of dehiscence and protection. Dehiscence and protection agilla andre pollen grains release agalla. Okay? Ill nodi students, ide nanu helidu sporogenous tissue. ಈ ಟಿಶ್ಯೂ ಬಂದ್ಬಿಟ್ಟು ಮೈಕ್ರೋಸ್ಪೊರಾಂಜಿಯಂ ಸೆಂಟರ್ ಅಲ್ಲಿ ಇರುತ್ತೆ. ಓಕೆ, ಲೈಕ್ ದಟ್ ಸೆಂಟರ್ ಸ್ಟಾರ್. ಓಕೆ, ಡಾನ್ಸ್ ಅಲ್ಲಿ ಒಂದು ಸೆಂಟರ್ ಪರ್ಸನ್ ವಿಲ್ ಬಿ ದೇರ್, ನೋ? So like that, one center it will be present, this porogenous tissue. Our tissue get potentially it can become the pollen mother cell. Let's take example. Okay, we have Uday in the class. I'm telling Uday, you can become a class leader. Okay, andre you know, I have seen Uday's potential and I'm telling that Uday ninage agbodu. Okay, ನೀನು ಆಗಬೇಕು ಅಂತ ನಾನು ಫುಲ್ ಪ್ರೆಷರ್ ಕೊಡ್ತಾ ಇದ್ದೀನಿ ದಟ್ ಯು ಕ್ಯಾನ್ ಡೂ ಇಟ್. ವೈ? ಬಿಕಾಸ್ ಐ ನೋ ಉದಯ್ಸ್ ಪೊಟೆನ್ಷಿಯಲ್. ಆ ಥರನೇ ಈ ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಕ್ಯಾನ್ ಪೊಟೆನ್ಷಿಯಲಿ ಬಿಕಮ್ ದ ಪೋಲನ್ ಮದರ್ ಸೆಲ್. Pollen mother cell and they are going to do the microsporogenesis. Why? So ultimately we can get the pollen grains, and inside the pollen grains there is going to be the male gamete. Okay, so you'll notice this porogenous tissue, students, they are potentially going to become the pollen mother cell or the microspore mother cell. But now microspore mother cells ge MMC anta bariyalla, because megaspore mother cell MMC ne. ಓಕೆ, ಸೊ ಅದನ್ನ ಕನ್ಫ್ಯೂಸ್ ಆಗಬಾರ್ದಲ್ವಾ, ಅದಕ್ಕೆ ಮೈಕ್ರೋಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ಸ್ ಅಂತ ಬರೀರಿ, ಇಲ್ಲಾಂದ್ರೆ ಪೋಲನ್ ಮದರ್ ಸೆಲ್ಸ್ ಅಂತ ಬರೀರಿ. ಅಂಡರ್ಲೈನ್ ಮಾಡಿ ಬಿಫೋರ್ ರೈಟಿಂಗ್ ಇಟ್ ಇನ್ ದ ಎಕ್ಸಾಮ್. ಕ್ಲಿಯರ್ ಟು ಆಲ್ ಆಫ್ ಯು? ಯೆಸ್ or no? Okay, now we will try and understand. So NCERT snippu ಹಾಕಿದ್ದೀನಿ so that you can understand the explanation. Now look here, what is microsporogenesis? This is one of the most important concepts of your topic. Microsporogenesis means formation of microspores from microspore mother cell or pollen mother cell through meiosis. Through what? Through meiosis, reductional division. Okay, through meiosis. ಇಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, ಏನಾಗುತ್ತೆ, ಸ್ಪೋರೋಜಿನಸ್ ಟಿಶ್ಯೂ ಇತ್ತಲ್ವಾ ಸೆಂಟರ್ ಅಲ್ಲಿ, that tissues are going to undergo meiosis, which is a reductional division, and they are going to form microspore tetrad. Tetrad andre ಏನು ಮ್ಯಾಮ್? 4 ಮ್ಯಾಮ್. Tetra means 4, tetra pack. Okay, tetrad means 4. So 4 microspores are formed. First 4 microspores ಬಂದ್ಬಿಟ್ಟು ಒಟ್ಟಿಗೆ ಇರುತ್ತೆ. ಓಕೆ, ದೇ ವಿಲ್ ನಾಟ್ ಬಿ ರೆಡಿ ಟು ಲೀವ್ ಈಚ್ ಅದರ್. ಅದಾದಮೇಲೆ ಒಂದು ಎನ್ಸೈಮ್ ಯೂಸ್ ಮಾಡಿಬಿಟ್ಟು ಅದನ್ನು ಸ್ಪ್ಲಿಟ್ ಮಾಡ್ತೀವಿ. ಸೊ ಸ್ಪ್ಲಿಟ್ ಆದ ಟೈಮಲ್ಲಿ ನೋಡಿ ಸ್ಟೂಡೆಂಟ್ಸ್, 1 2 3 4 ಇದೆಲ್ಲ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಇರುತ್ತೆ. ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಅಂದರೆ ಜಸ್ಟ್ N, ಡಿಪ್ಲಾಯ್ಡ್ ಅಂದರೆ 2N. ಓಕೆ, ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಇರುತ್ತೆ. Now each of the microspore tetrads can become a pollen grain. Okay, what did I say? What did I say? Each of the microspore can become the pollen grain. Correct? Yes. Why Karnataka students won't put thumbs up? They won't put thumbs up. They won't be active in the class. In students? Huh? ಬೇಗ ಎಲ್ಲರೂ ವಿಡಿಯೋನ ಲೈಕ್ ಮಾಡಿ ಬೇಗ. ಓಕೆ, 17 votes ಬಂದಿದೆ, ಬಟ್ ನೋಡಿ ನೀವು ಯಾರು ಅಪ್ರಿಷಿಯೇಟ್ ಮಾಡ್ತಾ ಇಲ್ಲ. ಫಸ್ಟ್ ಯೂನಿಟ್ ಟೆಸ್ಟ್ ಇಸ್ ಸೋ ಇಂಪಾರ್ಟೆಂಟ್. ಎಸ್, ಓಕೆ, ಚಲೋ, ಅಂಡರ್ಸ್ಟುಡ್. Now look here, this is your each of the microspore. Okay, students, each microspore can become the pollen grains. Okay, why do we need to form the pollen grains? Because ma'am, pollen grains olgane pollen male gamete irodu. Okay, now sac of pollen grains ಗೆ ಏನು ಕರೀತೀರಾ? pollen sac ಅಂತ ಕರೀತೀರಾ ಅಲ್ವಾ? Simple. ಈ ಗ್ಲಾಸ್ ಅಲ್ಲಿ ಇವಾಗ let's imagine in this glass, let's say there is lipsticks. So you will tell this is my lipstick pouch. Let's say in this glass there is pencil. So you will tell, yeah, this is my pencil pouch. Let's say in this glass you have pen. So you will tell, yes, it's my pen pouch, or let's say stationery pouch. So whatever is inside this, depending on that, we will be calling it. Before, sporogenous tissues were present inside it, so we call it what? Microsporangium. Now pollen grains are inside it, so we call it what? Pollen sacs. Okay, yes. Yes, pollen grain is the gametophyte. Okay, it is the gametophyte which is going to have the male gamete. Male gamete obviously haploid irutte alva. So idu haploid agide nodi, idu haploid agirutte, so it will be the same. Okay, now look here, see, each of the microspore will undergo mitosis. Each of the microspore will undergo mitosis and it will form the entire pollen grain. Pollen grain structure is very, very important. ಇಲ್ಲಿ ನೋಡಿ ಏನಾಗುತ್ತೆ. ಸೊ ಸ್ಟೂಡೆಂಟ್ಸ್, ನಿಮ್ಮ NCERT ಅಲ್ಲಿ ವ್ಯಾಕ್ಯುಲೈಸೇಷನ್, ಸ್ಪಿಂಡಲ್ ಫಾರ್ಮೇಷನ್ ಎಲ್ಲ ಕೊಟ್ಟಿದ್ದಾರೆ, ಬಟ್ ಇಟ್ಸ್ ನಾಟ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಫಾರ್ ಯು ಟು ಎಕ್ಸ್ಪ್ಲೇನ್. ಬಟ್ ಇಲ್ಲಿ ನೋಡಿ, ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ outermost layer is called the exine. external, outside, exine, okay? Then innermost layer is going to be called the intine. Now, exine is made up of sporopollenin. Is sporopollenin banbittu one of the most resistant substance ever. Okay, most resistant substance ever. Why? Because this fellow will not be affected by acid, alkali, temperature. Neeven madreno, it will be like, I don't care. Okay, just like how our mothers tell, no, whatever I scold you, you will not understand. You have buffalo skin. Kelidira maneyalli? Yen heldreno, ninaga artha agalla. Okay? So that is same thing here, sporopollenin. You put acid, you put alkali, you put temperature, it will be like, I don't care. Because I am the most resistant substance in the world, okay? And we have not found anything that would degrade this sporopollenin. Because of which, students, the pollen grains transfer agi protect mada job exine will take care of. Next one, you have intine, which is made up of cellulose and pectin. Okay, intine is made up of cellulose and pectin. Now, if you look at exine, you will see that there is some space that I have left. Because, students, it is the hardest, most resistant substance. ತುಂಬಾ ಡಿಫಿಕಲ್ಟ್ ಆಗುತ್ತೆ ಪೋಲನ್ ಟ್ಯೂಬ್ ಜರ್ಮಿನೇಷನ್ ಆಗಕ್ಕೆ. ಪೋಲನ್ ಟ್ಯೂಬ್ ಜರ್ಮಿನೇಷನ್ ಆಗಬೇಕಂದ್ರೆ ಒಂದು ಸ್ಮಾಲ್ ಸ್ಪೇಸ್ ಇರಬೇಕಲ್ವಾ? ಆ ಸ್ಪೇಸಿಗೆ ಸ್ಪೋರೋಪಾಲಿನ್ ವಿಲ್ ಬಿ ಆಬ್ಸೆಂಟ್ ಇನ್ ಒನ್ ಪೋರ್ಷನ್ ಅಂಡ್ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ದ germ pore. What is that called? The germ pore. Clear? Now, coming into the cell, you will see the pollen grains is split into two cells. One bigger portion and one smaller portion. Now, this bigger portion is called the vegetative cell. Vegetative cell, they are going to provide the nourishment for the male gamete. Where is the male gamete? It is going to be in the generative cell. Male gamete is going to be in the generative cell. So, we have generative cell, we have vegetative cell. Generative cell will have round nucleus, vegetative cell will have round nucleus, generative cell will have spindle shaped nucleus. ಇದೆಲ್ಲ ನೀವು NCERT ಅಲ್ಲಿ explain ಮಾಡಿದ್ದಾರೆ line to line. Okay, line to line. Clear? Yes or no? Is this clear? So we understood that the entire pollen grains has an exine, has an intine. Exine is made up of sporopollenin, intine is made up of cellulose and pectin. The entire pollen grains has two parts, one vegetative cell, one generative cell. Vegetative cell has round nucleus, more cytoplasm. Why? Because it has the food reserve for the male gamete. On the other side, you have generative cell, which is smaller, which floats in the generative cell cytoplasm, and it has a spindle-shaped nucleus. Okay, it has a spindle-shaped nucleus. Now, why are tapetal cells binucleated? Anta one question kelidare. So, main reason ಏನಂದ್ರೆ students, tapetal cells undergo karyokinesis. Karyokinesis ಅಂದ್ರೆ nucleus divide ಆಗುತ್ತೆ. But nucleus divide ಆದ್ಮೇಲೆ normally ಏನಾಗುತ್ತೆ? Cell wall formation ಆಗುತ್ತೆ ಇಲ್ಲಿ. Okay, cytokinesis follow ಮಾಡಬೇಕಾಯಿತು, but ಅದಾಗಿಲ್ಲ, because of which tapetal cells are binucleated. That means karyokinesis not followed by cytokinesis. If you want, you can remember. If it is too difficult, you can skip. Okay, you can skip. So, ಇಲ್ಲಿ NCERT highlight ಮಾಡಿದ್ದೀನಿ so that you know it will be easy for you people to study. So, go through NCERT. Don't miss NCERT. Okay. Okay. Now, look here. In your NCERT, they have mentioned in over 60% of the angiosperms, pollen grains are shed at two-celled stage. Now, what is this two-celled stage? Okay, look here. Two-celled stage means we have one vegetative cell and one generative cell. Normal 60% angiospermally. Okay, 60% angiospermally, one vegetative cell irutte, one generative cell irutte. They will dehiscent and normal everything will happen. Okay, but in other 40% alli, three cell stage irutte. One vegetative cell, two generative cell. Two generative cell artha eno? Two male gametes. Okay, two male gametes. Whenever two generative cell we are talking about, that means we are talking about two male gametes. Okay, this is in what stage? It is in three-celled stage. Okay. No, dear. No. Ornamentation of the pollen grains depend on how the sporopollenin lining is there. Pollen grains are used for different ornamental purpose, and if you look under the microscope, you will notice that each pollen grains are different because of how the sporopollenin arrangement is there. But otherwise, nothing like that. Okay. Yes. Now, moving forward. So, two-cell stage, three-cell stage, artha aitalva. So, what is two-cell stage? Two-cell stage andre 60% angiosperm alli enagutte? Pollen mother cell meiosis ಮಾಡಿ, we will form 4 microspore tetrad. Each of them will undergo mitosis to form pollen grain. So, ಇದರಲ್ಲಿ ಏನಾಗುತ್ತೆ 1 meiosis ಮತ್ತೆ 1 mitosis ಆಗುತ್ತೆ. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ 3 cell stage ಅಲ್ಲಿ ಇಷ್ಟು ಡೀಟೇಲ್ ಆಗಿ ನಿಮಗೆ ಲೆಕ್ಚರ್ ಎಲ್ಲೂ ಸಿಕ್ಕಲ್ಲ. So that's why I'm telling, please watch this lecture before going for your unit test. Okay, this lecture has everything in detail, including the questions that are in between your NCERT lines. Okay, in text questions illi nodi, in text questions ide. So that's why I'm telling you this lecture is enough. So please watch the lecture till the end. Now look here, 3 cell stage students, 40% of angiosperm, pollen mother cell will undergo meiosis and it will form microspore tetrad. Each microspore tetrad can become a pollen grain. I already explained madidini. So each of them will do mitosis. So normally on pollen grain alli ಏನು ಇರುತ್ತೆ, one vegetative cell, one generative cell. But ಈ generative cell halfway ಅಲ್ಲಿ he'll be like, yeah, one male gamete, say what will I do? I want 2 male gametes. So what he will do? He'll undergo one more mitosis and we will get 1 VC and 2 GC. Andre 1 vegetative cell, 2 generative cell, making it a 3 celled stage. So ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ students, 1 meiosis ಮತ್ತೆ 2 mitosis. Okay, 1 meiosis ಮತ್ತೆ 2 mitosis. ಅರ್ಥ ಆಯ್ತಾ ಎಲ್ಲರಿಗೂ? Did you all understand? Please give me a thumbs up. That much you can do students. Come on. Please give me a thumbs up. Quickly. Yes, faster. Yes, now the diagram in NCERT makes sense. Awesome. Okay. Yes. So, illenagutte, understood, 1 meiosis matte 2 mitosis agide. This is what happens in 3 cell stage. Now, pollen grain allergy anta on topic kottidare. Most of you will know. See students, very simple. Okay, ನೀವು ಯಾರಿಗಾದರೂ ಫ್ಲವರ್ ಕೊಡಬೇಕಂದ್ರೆ ಅವ್ರಿಗೆ pollen allergy ಇದೆಯಾ ಇಲ್ವಾ ಅಂತ ಕೇಳ್ಬಿಟ್ಟು ಫ್ಲವರ್ ಕೊಡಬೇಕು, ಇಲ್ಲಾಂದ್ರೆ you are going to get punched on your face. Why? Because there are people who get extreme pollen allergy. ಅವ್ರ ಬಾಡಿ ಫುಲ್ rashes ಬರುತ್ತೆ, ಅವ್ರಿಗೆ ಇನ್ಫ್ಲಮೇಷನ್ ಬರುತ್ತೆ, ಬ್ರೀತಿಂಗ್ ಇಶ್ಯೂಸ್ ಆಗುತ್ತೆ. So that's why you should know that if people have pollen allergy or not. Pollen allergy ಅಂದ್ರೆ ಏನು? Pollen grains ಅಲ್ವಾ, that will enter into the nostrils. It will directly go into your respiratory system. So adu, because of that, the entire body will start having inflammations and rashes. So due to pollen dust, sneezing, bronchial issues and rashes are going to come. One example given in your NCERT is Parthenium or carrot grass. Students, ಇವaglu carrot grass irutte nim area dal nodi. In case full city side illa andre ee tara on grass nodutte ನೀವು. Check this grass out. That is called as a Parthenium or your carrot grass. Now, viability. Viability topic ಬಂದ್ಬಿಟ್ಟು ಸ್ಟೂಡೆಂಟ್ಸ್ ಗೆ ತುಂಬಾ ಕನ್ಫ್ಯೂಷನ್. ಅಯ್ಯೋ ಇದೇನು viability ಅರ್ಥ ಆಗ್ತಾ ಇಲ್ಲ. ಸಿಂಪಲ್, ನಮಗೆ ಒಂದು OTP ಬಂದರೆ ಎಷ್ಟು ಟೈಮ್ ಆಗುತ್ತೆ ಅದರದ್ದು ವ್ಯಾಲಿಡಿಟಿ? ಲೈಕ್ ಅಂದರೆ ವಿತಿನ್ 5 minutes ನಾವು OTP ಎಂಟರ್ ಮಾಡಬೇಕು, ಇಲ್ಲಾಂದರೆ ಒಂದು 1 minute OTP ಎಂಟರ್ ಮಾಡಬೇಕಂತ ಇರುತ್ತೆ. ಬ್ಯಾಂಕಿಂದ ಬಂದರೆ 1 minute ಇರುತ್ತೆ, ಇಲ್ಲಾಂದರೆ ಒಂದು ನೈಕಾ, ಪರ್ಪಲ್, ಝೊಮ್ಯಾಟೊ, ಸ್ವಿಗ್ಗಿ ಆ ಥರ ಇದ್ದರೆ 5 minutes ಇರುತ್ತೆ. ಸೊ ವಾಟ್ ಇಸ್ ಗೋನ್ ಟು ಹ್ಯಾಪನ್? That OTP will expire after 5 minutes. After 5 minutes, if you enter the OTP, you will have to ask resend OTP. Correct? So here also students, after the pollen grains, after the dehiscence, let's say pollen grain has been taken by the wind, water, insect, anything. It has to go and fall on the stigma and it has to start the germination. Illa andre pollen grains expiry irutte. Andre pollen grains are not going to be active after that. So that is called as the viability. Just like OTP, here you have pollen viability. That means after releasing, the pollen grain has to go and land within this time for different species. So look here, viability gets affected by 2 factors. Andre viability can be affected depending on 2 external factors. One is humidity, okay, one is humidity, one is temperature. Now in cereals like rice and wheat, 30 minutes students, 30 minutes, within 30 minutes the pollen grain has to land on the stigma. Illandre adu viability completely destroy agutte. Now in families like Rosole andre Rosaceae family, Solanaceae family, Leguminosae family, the viability is going to be for months. Viability is going to be for months. Okay. Now students, idon doddu problem alva. Farmers ige they want, like you know, they want to do fertilization, they want to do artificial fertilization or aa tara 100 techniques ide. Now if pollen grains are not going to be viable, ತುಂಬಾ ಪ್ರಾಬ್ಲಮ್ ಆಗುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಒಂದು ಪ್ರಿಸರ್ವೇಷನ್ ಟೆಕ್ನಿಕ್ ಇದೆ. ಜಸ್ಟ್ ಲೈಕ್ ಹೌ ವಿ ಪ್ರಿಸರ್ವ್ ಅವರ್ ಬ್ಲಡ್, ಹೌ ವಿ ಪ್ರಿಸರ್ವ್ ಸ್ಪರ್ಮ್, ಎಗ್, ಆ ಥರ ಇಲ್ಲೂ ಒಂದು ಪ್ರಿಸರ್ವೇಷನ್ ಇದೆ. ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಕ್ರಯೋ ಪ್ರಿಸರ್ವೇಷನ್. Cryopreservation andre we can preserve the pollen grains at minus 196 degrees Celsius in liquid nitrogen. We can what? We can preserve tell me. Okay, so here preserve the pollen grains in minus 196 degrees Celsius in liquid nitrogen. So by this way what will happen students, namage pollen grains na preserve ಮಾಡಬಹುದು, later use ಮಾಡಬಹುದು, so that viability problem tension iralla. Okay, viability problem ಮತ್ತೆ tension iralla. Understood so far? Yes or no? So ಇಲ್ಲಿ ನೋಡಿ, you will understand exactly all the things that you are, it's very, very important. Haploid structures, okay? I think YouTube channel, I think that's the name, no? You were asking. So look here, these are formed after meiosis, contains only one set of chromosome, and just N, okay? Only N is present. What are they? Microspores. Microspores are the ones after meiosis, because we did reductional division, so we got microspores. Microspores in the we got pollen grains, so obviously adu banbit haploid agirutte. Matte vegetative cell and generative cell. So pollen grains completely haploid andre adu whatever is inside the cell also will be haploid only. Okay, can we somehow reverse the expiry beside? No, we cannot. We cannot. Okay, now gametes. Okay, gametes also, that is your sperm cells or your male gamete, and the female gamete is going to be haploid. Why? Because fertilizationally n plus n gives you 2n. So idu diploid agutte alva. So zygote is going to be diploid. So if zygote has to be diploid, then we need our sperm and egg, or the male and the female gamete, to be diploid, correct? So here your zygote will be diploid, but the male and the female is going to be haploid. Now diploid structures nodi, pollen mother cell matte megaspore mother, microspore mother cell. Why? Because avrige meiosis agbeku. Okay, reductional division ಆಗಬೇಕು. Reductional division ಆಗಬೇಕಂದ್ರೆ MMC ಮತ್ತೆ PMC ಬಂದ್ಬಿಟ್ಟು ಡಿಪ್ಲಾಯ್ಡ್ ಆಗಿರಬೇಕು. So diploid undergoes meiosis. Meiosis ಅಂದ್ರೆ ಏನು ಸ್ಟೂಡೆಂಟ್ಸ್? Reductional division. Okay, reductional division. Clear? Now, sporogenous tissue. Sporogenous tissue give rise to PMC. Okay, it has to give rise to the mother cell. So obviously it also it has to be diploid. So sporogenous tissue also is diploid. Walls of anther, epidermis, endothelium, middle layer, tapetum, ella structurally diploid agirutte because they surround the sporogenous tissue. So ee confusion completely avoid ಮಾಡಿ. Microspores ಬಂದ್ಬಿಟ್ಟು haploid ಆಗಿರುತ್ತೆ, pollen grains haploid ಆಗಿರುತ್ತೆ. Pollen grains ಒಳಗಡೆ ಇರೋ vegetative cell ಮತ್ತೆ generative cell divide, I am going to get n plus n, right? Half. Reductional division is going to happen. Clear? Viability also is clear, no? When once the pollen grains are shed, pollen grains have to land on the stigma before they lose the viability if they have to bring about fertilization. Fertilization agbekandre they have to land within the time that is mentioned. Illi nodi cereals adre rice and wheat ge 30 minutes, Rosaceae family, Solanaceae family, Leguminosae family ge months. Okay, months time is there. Months time is there. And here we can preserve them. Okay, we can preserve them by doing cryopreservation, that is at minus 196 degrees Celsius in liquid nitrogen. Clear? Yes or no? Okay. FA1 alva, FA4 ah. Okay, pistil matte carpel. So students, male portion completely over. Okay, what and all did we study about androecium that you have to remember? Okay. So this is like GM Express, okay, Gopika Ma'am Express. So here we studied everything about androecium, yes or no? Okay, androecium. Students, androecium is made up of units called stamen. What is stamen made up of? Anther and filament. Okay, anther and filament. Anther is made up of what? And sorry, anther is made up of alla. Anther characteristics eno, it is bilobed. Okay, 2 lobes ide. Dithecous, 1 lobe has 2 thecus, so dithecous. tetrasporangiate. Okay, tetrasporangiate. Okay, tetrasporangiate. So these are the things that you have to remember about your anther. Okay, filament one but nothing, just long and slender. Okay, long and slender. So androecium is the essential part. So since androecium is the essential part, these are going to help in the formation of the male gamete. Okay, MG I'm writing. So male gamete. Male gamete formation henga agutte with the help of microsporogenesis, which can come for your FA1 exam, because microsporogenesis usually keltare. Okay, microsporogenesis andre en students, formation of microspores. Okay, formation of microspores with the help of meiosis. From what? From PMC. Okay, PMC illandre microspore mother cell will undergo meiosis. Okay, will undergo meiosis and they will form microspore tetrad. Each microspore tetrad will form pollen grains by undergoing mitosis. Okay, they are undergoing mitosis, that is why both are N and N. Before mitosis will happen, this pollen grain, what and all they have? They have exine, which is made up of sporopollen. They have intine, which is made up of cellulose. And so VC and GC is there. In the GC, MG is going to be there. That is the male gamete. Okay, 3-cell stage can be there. Okay, which is seen in 40% angiosperm. 2-cell stage can be there, which is seen in 60% of angiosperm. Okay, now pollen allergy. Pollen allergy. Ee tara on paper ella bardiktre, it will be easy for revision. Okay, pollen allergy, students, it is going to be Parthenium grass. Okay, Parthenium, which is called the carrot grass. Okay, Parthenium, which is a carrot grass. And then you talk about viability. Okay, ha ma'am, FA4, FA1, 2, 3 completed in May and June. Now FA4 portions are also complete. Oh, Indumati, which college are you in? It's going in helicopter speed. Huh? Okay. Now, coming back here, okay, here we are going to start with the male, sorry, female, okay, female we are going to start. So, you'll notice students, stigma style ovary matte ovules, idella form agadeno, this is the pistil or illandre carpel. This, all the units are present inside what? Gynoecium, okay, gynoecium. So, it's all formed in the gynoecium. So, G for girl, G for gynoecium, like that you can remember. You'll notice stigma style ovary ovules, which will form the pistil and carpel. Now, carpels can be depending on two. Okay, there can be two types: monocarpellary matte multicarpellary. Okay, monocarpellary matte multicarpellary. Monocarpellary andre single. Mono andre okay, they'll like okay, single, one person is there. Multicarpellary andre they will be having many carpels. Okay, neevellaru hibiscus plant. Okay, the hibiscus plant illandre China rose anta karitivi. You go take that and you will be able to see what? You will be able to see that these people have multi carpels. Okay, many carpels. Now students, many carpels idre, let's say you have many friends. Okay, it is up to you if you want to be free from them or if you want to be fused to them. Fused to them, okay, wherever they go, you will go. Wherever they party, you will party. Whatever they tell, you will also agree to this. But there are friendships where you will be like, no, yaar, I am not coming for this. I don't agree with this. So you can be free also in a friend circle or you can be fused also. ಆ ತರನೇ ಮಲ್ಟಿಕಾರ್ಪುಲರಿಯಲ್ಲಿ ಅಪೋಕಾರ್ಪಸ್ ಮತ್ತೆ ಸಿನ್ಕಾರ್ಪಸ್ ಇರುತ್ತೆ. ಅಪೋಕಾರ್ಪಸ್ ಅಂದ್ರೆ ಫ್ರೀ ಆಗಿರುತ್ತೆ. ಇದರದ ಎಕ್ಸಾಂಪಲ್ ಯು ವಿಲ್ ರಿಮೆಂಬರ್ ಮೀ, ಮ್ಯಾಮ್. ಓಕೆ, ಓಕೆ ಓಕೆ ಇಂದುಮತಿ, ಅಂಡರ್ಸ್ಟುಡ್. ಓಕೆ, ಮ್ಯಾಮ್. M for multicarpellary, A for apocarpus. Example ಬಂದ್ಬಿಟ್ಟು ಮಿಶೆಲಿಯಾ. ಓಕೆ, MCQ ಕೇಳ್ಬೋದು, ಇಲ್ಲಾಂದ್ರೆ ಇಟ್ಸ್ ಫೈನ್. ಓಕೆ, ಸಿನ್ಕಾರ್ಪಸ್ ಅಂದ್ರೆ ಜಾಯಿಂಟ್, ಫ್ಯೂಸ್ಡ್ ಆಗಿರುತ್ತೆ. ಸೊ ಯು ರಿಮೆಂಬರ್ MSP, multicarpellary, syncarpus and papaver. Okay, papaver. So that is what is given here. ನೋಡಿ, ಇಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ಕೊಟ್ಟಿದ್ದಾರೆ. NCERT ಅಲ್ಲಿ ಎಲ್ಲೆಲ್ಲ ಸೈಡಲ್ಲಿ ಏನೆಲ್ಲ ಬರ್ದಿದೆ ಅಂತ ನಿಮ್ಗೆ ಗೊತ್ತಿರಬೇಕು. ಓಕೆ, yes. Now we will study about the ovule. Okay, we will study about the ovule. Yes, so far understood everything? Okay. Understood? Yes, tell me quickly. Okay. Okay. Now let's do ovule. Okay? Anatropous ovule. So students, the ovule ನೋಡ್ಬಿಟ್ಟು ನಿಮಗೆ ಅರ್ಥ ಆಗುತ್ತೆ ಮ್ಯಾಮ್ ಇದೇನು ಮ್ಯಾಮ್ ಫುಲ್ ಅಪ್ಸೈಡ್ ಡೌನ್ ಆಗಿ ಡ್ರಾ ಮಾಡಿದ್ದೀರಾ. So that is your anatropous ovule. Now what is anatropous ovule means? Students, look here. This is the funicle. This is the chalazal end. This is the micropylar end. They're not coming in a straight line. Right? They're not coming in a straight line. So that is what we are telling here. Here you see this chalazal end. Okay? Chalazal. So here this portion is the chalazal end, which is actually considered as the base of the ovule. Base of the ovule. Illi nodi, don't break your head. First ಏನಿರುತ್ತೆ ಈ ಎರಡು ಲೇಯರ್ಸ್ ಕಾಣಿಸ್ತಾ ಇಲ್ವಾ? These two layers are called as integuments. Integuments ಅಂದ್ರೆ ಏನು? Protection. For protection we have integuments. Clear? Okay? Integuments. Now look here. These two are the integuments. So we have inner integument and outer integument. What are they going to do? Protection. What are they going to do? Protection. Okay? Adamel students, illon portion nodi integuments absent agide. Absent agide. That portion is called as the micropyle. Region where integuments are absent. All these notes will be available on Vedantu Karnataka Telegram. If you want to join my Telegram, it's Biology, Gopika Biology Official. You can also follow me on Instagram. Okay? So micropyle. Now this stalk-like structure which is holding the entire ovule together is called the funicle. Okay, it is called the funicle. Now funicle is attached to the ovule in a portion which is called the hilum. What is it called? Hilum. Kidney allo on hilum ide, ad jote confuse agbedi. So funicle is a stalk to like funicle is attached to the ovule at one portion that is called the hilum. Okay, that portion that is called the hilum. So that connects funicle and ovule. Now students, look here, in the middle what you are seeing, no, pink color, that is called the embryo sac. That's called the embryo sac, which is the female gametophyte. So all ಏನito pollen grains ಬಂದ್ಬಿಟ್ಟು male gametophyte ಆಗಿತ್ತು. ಇಲ್ಲೇನೋ embryo sac female gametophyte. ಅಂದ್ರೆ male gametophyte ಒಳಗಡೆ male gamete ಇರುತ್ತೆ, female gametophyte inside female gamete will be there. So this is the embryo sac. And can you see there's one layer of cell which is surrounding the embryo sac, which is called the nucellus. Now nucellus, not nuisance, okay, nucellus, that provides nourishment for the growing embryo sac. But on twist ide nodi illi. Okay, so this is understood. So last time we explained madidini. Nucellus, so one of the nucellus cell, okay, in our class also all of you would have noticed there'll be one, what is that, very over smart boy or girl. They'll be like, ma'am, I know the answer. Ma'am, I'll go take the book. Ma'am, assignment you gave yesterday. Ma'am, homework you didn't give. You gave homework yesterday. You did not check the homework. We all used to like, or we used to all dislike that student. Yes or no? Now, homework madidre, then it's fine. If we didn't do the homework, if the student tells that, yeah, hi, okay, ma'am, you gave homework and, you did not check, you would be so angry at the student, no? Just like that, one of the nucellus cells from the micropylar end, okay? One of the nucellus cell from the micropylar end will be like, I will become the megaspore mother cell. I have the capabilities, I will become the megaspore mother cell and it will form the megaspore mother cell. So already explained madidini, megaspore mother cell bandu diploid agirutte because they are going through a reductional division, not an equational division. So megaspore mother cell 2n, this megaspore mother cell will undergo meiosis and same microsporogenesis tarane illu copy agutte, 4 megaspores form agutte. Okay, 1 2 3 4, but unfortunate. Okay, unfortunate. Oh, Bassa sir is in the chat box. Okay, so what is going to happen? What is going to happen here? 1 2 3 4 are there, no? In that 3 of them are going to degenerate. 3 of them are going to become non-functional. Only one will be like, okay, wait, I will do the continue. I will continue from here. Don't worry about it. So illenaitu, nucellus cells were covering the embryo sac. Adinda one nucellus cell, potential nucellus cell will be like, I will become the megaspore mother cell. Megaspore mother cell will undergo meiosis. Meiosis in the, we are going to get a megaspore tetrad. But unfortunately, 3 of them will degenerate and only one will become functional. Since only one is becoming functional, we call that development as monosporic development. Mono andre single, spore andre one spore. So only one of them is becoming functional. Understood? Yes or no? So monosporic development. Now students, idal nodi, this one fellow will become 2, okay, like division will happen, okay, 1 to 2, 2 to 4, 4 to 8. By undergoing 3 mitotic divisions, they are going to form your embryo sac. Inside embryo sac, what will be there? Female gamete is going to be there, okay, female gamete is going to be there. Understood? So ಇಲ್ಲಿ ನೋಡಿ explanation same ಕೊಟ್ಟಿದ್ದಾರೆ, funicle, hilum, integument, chalazal, micropyle, basal part of the ovule. Understood? Now look here, enclosed within the integument is the mass of cell called the nucellus. Okay, important. Cells of the nucellus have abundant food reserve material. Located in the nucellus is the embryo sac or the female gametophyte. Megasporogenesis, the process of formation of megaspore from megaspore mother cell is called as megasporogenesis. Understood? So illenagutte ee diagram nodi, ee diagram indane explain martini. All of you have your NCERT in front of you. So look here, you can see the megaspore mother cell. Okay, this is the megaspore mother cell. They will undergo division to form megaspore dyad. From megaspore dyad, they will form megaspore tetrad. But ನಾನು already explained ಮಾಡಿದ್ದೀನಿ, the megaspore tetrad, 3 of them will become non-functional. Okay, 3 of them will become non-functional or degenerate, and only 1 will become functional. So that 1 will go from dyad, okay, from dyad stage 2 to 4, 4 to 8 stage. That 8th stage is going to align themselves and form the embryo sac. So let's imagine, students, I am putting all of you in one classroom. Okay, let's take example. Okay, I have here Sunita, I have here, okay, Aishu, I have here, who is there? I can't see your name. Prakash is there. Okay, here let's say I have, you know, few of them. Let's say here also I have few of them. Okay, like this, 8 students are there in the class. Okay, 8 students are there in the class. Suddenly I'll tell them, on your mark, get set, go. So get set, go illa time alli, let's assume 3 jana ಇಲ್ಲಿ ಬಂದು ಕುತ್ಕೊಂಡರೆ. Okay, 2 people center, 3 people here. So that means what? 3 people have moved, okay, towards the pole, this pole, and 2 people in the center. 2 people in the center, just like that. Okay, Sanjana also is here. Okay, just like that, students. Illenagutte 8 cell stage alli our align agutte. 3 people will go towards the pole, another 3 people will go towards the micropylar end. So like that only here also. When I told on your mark, get set, go, let's say few of you went to the chalazal end. Okay, few of you went to the micropylar end. Understood? That is what happens. That is what happens. So illi finally you will be able to see the formation of an embryo sac, where you will notice that 3 cells, okay, there are 3 cells which are called as antipodal cells, which are towards the chalazal end, and you call that what? AC. Okay, remember like this, AC. AC gotala? Okay, AC. So antipodal cells towards the chalazal end. In center right, you will have your polar nuclei, and then this portion is called the central cell, and here you have the egg. So what is egg? The female gamete is the egg. Right? Female gamete is the egg. This entire thing is the female gametophyte. Okay? Female gametophyte. Students, gametophyte are bodies which are bearing the gametes. Okay? Bodies which are bearing the gamete. Female gametophyte. Understood? So like example, let's say, this glass is pen ide. Okay? This glass becomes the gametophyte, this pen becomes the gamete. Okay? I'm telling you, this is the gamete, this is the gametophyte. So this is pollen grain, so inside this male gamete will be there. Let's say this is embryo sac, inside that egg is going to be there. Understood? Okay? Understood? Clear? Clear, clear? Okay. Now, chalazal end, antipodal cells, polar nucleus, central cell, synergids, filiform, understood. Okay? Filiform, understood. Now, next topic that we have is pollination. How do these pollen grains go from, you know, how are they transferred? So, students, pollination andre normally, definition, transfer of pollen grains from anther to stigma, right? This is the anther, this is the stigma. So ee anther inda pollen grains scattered agi stigma mele bidutte. From there the germination will continue. But pollination is of 3 types. First one is autogamy, next is gaietnogamy, then it's xenogamy. Autogamy andre eno nam phone allu on setting ide auto mode andre light contrast what is that black point ella avare adjust ಮಾಡ್ತಾರೆ. Auto self driven. Okay, so autogamy andre self pollination, xenogamy andre cross pollination, gaietnogamy andre one type of self pollination. Okay, one type of self pollination. So ಇಲ್ಲಿ ನೋಡಿ autogamy andre ಏನು same flowers, that is pollen grains from the same flower is going to fall on the stigma of the same flower. So look here, illinda pollen grains ee stigma transfer ಆಗ್ತಾ ಇದೆ, everything is happening in the same flower. So transfer of pollen grains from the anther to the stigma of the same flower is called as autogamy. Okay, gaietnogamy one type of self pollination, but illondu ಸ್ವಲ್ಪ difference ಏನಂದ್ರೆ ಒಂದು flower ಇಂದ ಇನ್ನೊಂದು flower ಗೆ. Okay, just like how you have marriage. Okay, I don't know, in here it's very less, but Tamil Nadu it's very common. Mama ponnu, mama payyan, okay, atte ponnu, atte payyan, all that they get married. No, that is like inside the family only they'll get married, like to the aunt's son or daughter. So that means what? Here, whatever genetic disorders is been running in the family will continue in the family. Alva, change agalla because ashtu variation illa, ellaru same family ne, but it's not like, you know, you understand, no, it's like aunt and uncle's kids. So illu atarane onde plant alli different flowers, you know, madhyadalli agadu transfer of pollen grains between anther to the stigma of different flowers of the same plant. Correct ag baribeku, different flowers of the same plant. Okay, that is called as your gaietnogamy. Understood? Now, cross pollination from small age you are studying transfer of pollen grains from anther to stigma of a different plant, completely different plant. Okay, yes, gaietnogamy no similar genes irutte Prakash, because allu jaasti difference illa, variation illa, same family olagadene ನೀವು ಮದುವೆ ಮಾಡಿದ್ರೆ whatever genetic disorders are there is going to continue. There's no much change. Okay, there's no much variation that is going to come. Clear? Now, agents of pollination means you will have biotic and abiotic. Biotic means what? Biotic means you have insects living. Okay, insects will be there. Insects are going to be like honeybee is the most common one. We also have beetles, flies, butterflies, atara tumba ide alva. Illi nodi, insect students, insect is very smart. They know that okay, I am going to the flower and I am helping the flower to do pollination. So insects ಏನು ಮಾಡ್ತಾರೆ, they are going to get attracted because of the nectar and because of the fragrance of the flower. Now, this is your honeybee. Okay, at the same time there are people who get attracted to foul odor. Foul odor andre like a bad smell. Okay, bad smell, beetles and flies will get attracted to that. Abiotic andre water ಮತ್ತೆ wind. Water ಬಂದ್ಬಿಟ್ಟು ತುಂಬಾ rare students, because chances of pollen grains and you know egg meeting is very less. Wind ಬಂದ್ಬಿಟ್ಟು most common. So ಆ ತರ ಕೇಳ್ತಾರೆ which is the most common, which is the rare ಅಂತ. Okay, hi Praveen. Now this is what you have to study. Okay, my throat needs 1 minute break. Okay, by then you can just go through this. One second, students. They will ask you direct questions from this. Okay, so don't take the risk of skipping it. Okay, don't take the risk of skipping it, thinking that okay, I'll not study this, it's not that important. They can ask. Praveen, you watch my lecture, after that I'll make you do question practice, so you study that and go. Okay, it's more than enough. I know for some of you this chapter will be difficult, but it's okay students. Aram se you study, don't study like, you know, tension etkonbedi. Tumba ide ma'am, ayyo ishtella hengo odadanta. Okay? Chalo. Pollination by wind is more common amongst abiotic pollination. Abiotic andre non-living. biotic andre living. Okay, biotic andre living. Understood? So you look here, pollen grains are light. So students, very simple. Just imagine you want to clean. Okay, you want to clean. Let's say I have put very sticky slime everywhere. Okay, now if I put, okay, I will give you the PDF so you can revise from there, and other than that you can attend the class and practice the question. Let's say students, yella kadeno nan slime hakidini. Will it be easy for you to clean? No ma'am, it won't be easy to clean. But let's say same time you have some dry leaf. You will be like, ma'am, dry leaf is easy to clean because they will be moved very fast. Ill nodi, pollination by wind is more common amongst abiotic pollination. Wind pollination also requires pollen grains which are light and non-sticky. It has to be light and non-sticky, otherwise when the wind is trying to take them, it will not come, right? It will not come. So they can be transported in the wind currents. So directly nimage ee tara question keltare, what are the characteristics of wind pollinated flowers? So direct answer baribeku, pollen grains are non-sticky and lightweight so that it can be transported by wind. Now stigma and anther will be well exposed. So look here, they have large often feathery stigma. Large often feathery stigma to easily trap the airborne pollen grains. Wind pollinated flowers often have single ovule. Onde one ovule irado. Okay, one ovule will be there in each ovary and numerous flowers packed in an inflorescence. So students, just imagine madi, small flowers. Okay, itara small flowers ide, tumba kashta agutte pollen grains transfer madakidakke. So they will all form an inflorescence. Okay, they will all form an inflorescence. That means they will all cluster together. They will all cluster together and then they will look like one whole bunch of flowers. Okay, whole bunch of flowers. Clear to all of you? Now, this is the corn cob. Corn cob, ears do you see is nothing but the stigma and style. Okay? Yes, anemophily. Yes, anemophily we talk for wind, entomophily for insect, okay, hydrophily for water. Now, pollination by water is quite rare in flowering plants and it is limited to 30 genera. Okay, that means only 30 of them are going to do this water pollination. Tumba rare agide. Because students, illi niಮಗೆ ಗೊತ್ತು bryophyte ಮತ್ತೆ pteridophyte, ಅದರಲ್ಲಿ ನಾವು bryophyte ನ amphibians of the plant kingdom ಅಂತ ಕರಿತೀವಿ. Amphibians of the plant kingdom ಅಂದ್ರೆ ಏನು? ಅವ್ರ ಲೈಫ್ ಸೈಕಲ್ ಕಂಪ್ಲೀಟ್ ಮಾಡೋದಕ್ಕೆ ಅವ್ರಿಗೆ ನೀರು ಬೇಕು, ಲ್ಯಾಂಡು ಬೇಕು. So that is why bryophytes are not well furnished or they are not completely spread over because of the reason that they need water. But wherever water is there, they are there. Okay. So here, that is, water is very, like water pollination tumba rare agide. Ill few examples kottidare which are very important for you people. Look here, Vallisneria and Hydrilla which grow in fresh water and several marine sea grasses just like Zostera. Not all aquatic plants use water for pollination in majority. So Vallisneria matte Hydrilla water pollination ಮಾಡ್ತಾರೆ. That means they will come a little near the water surface and the pollen grains will be released. They will take in the pollen grains. But ಇಲ್ಲಿ ನೋಡಿ examples like water hyacinth ಮತ್ತೆ water lily. Water hyacinth ಮತ್ತೆ water lily ಆ ತರ ಮಾಡಲ್ಲ. ಅವ್ರು ಏನು ಮಾಡ್ತಾರೆ, they will come above the water level. They will wait for insect or wind to pollinate them. Because ಅವ್ರಿಗೆ ಗೊತ್ತು water pollination ಅಷ್ಟು ಕಾಮನ್ ಆಗಿಲ್ಲ, chances of pollination ಆಗೋದು ಸ್ವಲ್ಪ ಕಮ್ಮಿ ಇದೆ. ಸೊ ನಾನು ಯಾಕೆ ರಿಸ್ಕ್ ಮಾಡಬೇಕು, I will come above the water, let insect or wind pollinate. Okay, so look here, water hyacinth and water lily, the flower emerges above the water and are pollinated by insects or wind as in most of the land plants. In Vallisneria, the female flower reaches the surface of water by a long stalk and the male flower or the pollen grains are released onto the surface of water. So, water will carry them. Okay, water will carry the, water will carry the pollen grains and these pollen grains are going to come and, fertilization is going to happen. Clear? Yes or no? So, pollen grains are released to the surface of water. They are carried passively by water currents. Some of them eventually reach the female flower. Some of them eventually reach the female flower. Andre eno? Eventually reach the female flower andre, some of them might not even reach. Alva? Water will move ಆಗ್ತಾ ಇದೆ. So, there are chances ಅವ್ರು reach ಆಗಲ್ಲ. There are chances they will reach. So, ಆ ಥರ ಒಂದು problem ಇರುತ್ತೆ. Okay na? Clear? So, what will you write? For wind pollination, you will write the pollen grains are light and non-sticky. Stigma and anther are well exposed. Okay, they are feathery, so that too they can attract the pollen grains. Okay. Other than that, what will you write? They have single ovule. Flowers are not big. They are formed into an inflorescence. Okay. Clear? Yes. Understood? Clear? Okay, understood. Doubt? You have a doubt? What is Uday telling? Bere class ke baad sumne. Oh, they thought YouTube is WhatsApp. Yes. Clear? Done, done, done. All of you? Done? Okay. Okay, come on. Now, next is students, your insect pollinated. Insect pollinated flowers banbittu. Insects are like the, picky people. So, they will not just, they will not just go to any plants and do pollination. Our banbit most colorful plants, flowers yaudide, most nectar ones, fragrance ones, they will only choose flowers like that. So, here you see insect pollinated flowers, bees, butterflies, flies, beetle, wasps, ants, moths, birds, sunbirds and hummingbirds, and bats are common pollinating agents. Bees are the dominant biotic pollinating agents, okay. Even larger animals such as primates like a lemur or your tree dwelling rodents or reptiles, geckos, lizards, okay, they are all going to help in pollination. Now, what is the most specialized character? I already told you, they have to be large, colorful, fragrant, rich in nectar. Okay, so ella time insect bara time ali. Whenever insect is coming, the flower has to give insect a reward. Just like whenever you people come to the class, I tell you people like the video, comment on the class, that is the reward that I get. For being a teacher, for being active on YouTube, that is the reward that I want. Now, what is the reward that you get? Whenever I teach a class and you understand everything, you feel that confidence, yes, I understood it. And when you write really well in the exam, that is the reward that you get. So, whenever we are coming back for the reward system, whenever we are coming back to the class, we both expect the same. Correct? I expect a reward from you, you expect a reward from me. Same thing happens here. Every time the insect goes back to the flower is expected to give a reward to the plant, to the insect. Why? Because insect is going to help in pollination. So, ella time vapas hoga time ali flower has to give nectar, illandre pollen grains as a reward. Okay, pollen grains as a reward, and on behalf of that, insect is going to do pollination for them. So, look here, large, colorful, fragrant, rich in nectar flowers, cluster. If the flowers are too small, they will form a cluster called as inflorescence. Color or fragrance, the honeybees and all will get attracted to. Flies and beetles will get attracted to foul odors. Flowers have to provide rewards to the animals. Why? So that they can come back. That is going to be nectar and pollen grains are usually the floral rewards. Clear? Nectar and pollen grains are usually the floral rewards. Clear to all of you? This question, they will directly ask you what are the features of water pollinated flowers, what are wind pollinated flowers, what are insect pollinated flowers. Direct question, students. Okay, direct question. In Lodi, in some species, floral rewards are in providing safe places to lay eggs. An example of that is the tallest flower, that is Amorphophallus. So, students, ee flower alena some of the beetles will come and lay their eggs in the ovule. So, in Yavtara andre, let's take example. So, I told Praveen that I will teach you this chapter, I will give you the notes for it. So, Praveen will put the trust on me and he'll be like, okay, I'm not going to watch any other lecture, I'll wait for Gopika Ma'am to give me the notes or teach me this chapter. Now, that is what, it's a mutual understanding. Atarane, illenagute, ee Amorphophallus flower alli, some beetles will come and lay their egg in the ovules. Beetles will assume that, okay, this flower is going to protect the eggs. Instead, I will do for a pollination for the flower. Simple alva. So, these beetles are going to help in pollination of the flower. Flower is going to protect the eggs which are laid in the ovule. Atarane illi nimige yucca moth. Okay, yucca plant matte yucca moth illandre pronuba moth. So, avaru atarane this one you are seeing, no, this is the moth. This moth will lay eggs. Okay, this moth will lay eggs in the ovule of this plant and it will tell the plant, you please protect. Okay, instead what will this plant expect from this fellow? Plant will expect that this moth will pollinate. Okay, so we have some mutual symbiotic relationships. Understood all of you? Yes or no? So this is what happened. Yucca moth and Amorphophallus flower, which is almost a 6 feet tall flower. Done? So fly lay eggs in the flower, Yucca moth pollinates the flower, lays eggs in the ovary. Done? So this is how they are going to help each other. This is how they are going to help each other. Now, outbreeding devices. Outbreeding devices and they are very simple. ನೀವು ಕಂಟಿನ್ಯೂಸ್ ಆಗಿ ರೂಮಲ್ಲಿ ಕುತ್ಕೊಂಡ್ರೆ ನಿಮಗೆ ಡಿಪ್ರೆಷನ್ ಆಗುತ್ತೆ. ಯಾರ ಜೊತೆನೂ ಮಾತಾಡಲ್ಲ, ಯಾರ ಜೊತೆನೂ ಮೆಸೇಜ್ ಮಾಡಲ್ಲ, ಯು ಡೋಂಟ್ ಲಿಸನ್ ಟು ಮ್ಯೂಸಿಕ್, ಯು ಡೋಂಟ್ ವಾಚ್ ಮೂವೀಸ್, ಯು ಓನ್ಲಿ ಸಿಟ್ ಲೈಕ್ ದಿಸ್ and you'll be like, I will not talk to anyone. You're going to have depression. Same thing plants also go through. Because they do continuous pollination, but if they are continuously going to do self-pollination, they are going to have something called as inbreeding depression. What are they going to have? Inbreeding depression. Now students, to avoid inbreeding depression also, the plants have few tools. Okay, plants have few tools. Continued self-pollination leads to inbreeding depression. Clear? Now, the first way for the plant to avoid self-pollination henge? Flowers. Flowers na separate madodu. Unisexual flowers form madodu. Because male matte female, ಆಂಥರ್ ಮತ್ತು ಸ್ಟಿಗ್ಮಾ ಒಂದೇ ಪ್ಲಾಂಟಲ್ಲಿ ಇದ್ದರೆ ಪ್ರಾಬ್ಲಮ್ ಆಗಿರುತ್ತೆ, ಒಂದೇ ಫ್ಲವರಲ್ಲಿ ಇದ್ದರೆ ಪ್ರಾಬ್ಲಮ್ ಆಗುತ್ತೆ. ಸೊ ವಾಟ್ ವಿಲ್ ದೇ ಡೂ? ದೇ ವಿಲ್ ಫಾರ್ಮ್ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್. Unisexual flowers, that is staminate or pistillate flower. The ones that have stamens, the one that has pistils. Clear? Next one, asynchronized receptivity. ಅಂದರೆ let's say anther ಮತ್ತೆ stigma ಒಂದೇ ಟೈಮಲ್ಲಿ ಮೆಚೂರ್ ಆದರೆ ಪ್ರಾಬ್ಲಮ್ ಆಗುತ್ತೆ, because anther is going to put the pollen grains on the stigma. Stigma is going to continue the germination. So it will, let's say it will make the anther mature first, stigma mature later. Understood? So asynchronized receptivity, that means changing the maturity time of anther and stigma. Length of anther and stigma, ಇದು ತುಂಬಾ easy. ನೋಡಿ, stigma usually ಕೆಳಗಿರುತ್ತೆ, anther ಮೇಲೆ ಇರುತ್ತೆ, so that pollen grains can fall on the stigma. Instead of that, they made the stigma taller and anther shorter. So changing the length of the anther and the stigma. Last one with students, self-incompatibility. Plant will be like, okay, if my own pollen grains come and fall, I will reject them. Let's say my two pollen, this is one self pollen, this is from somewhere. Let's say two pollen falls. They will be like, no, reject mine. Only continue this. So this way what will happen? Inbreeding depression can be avoided. Understood? Self-incompatibility. Andre self pollen na completely reject ಮಾಡೋದು. Understood or not? Yes? Hi Manyashri. Okay, students, just a 5 minutes, okay, 2 minutes maybe. Okay, I'll just get some hot water and come. After that, we will do double fertilization. Okay, and then I will teach you about seed. Okay, I'll teach you about seed. Oh, it's repeat again, yeah. Okay. Students, JP Nagar, we have a center. So, if any of you are preparing for your KCET, NEET, JE, then you can join our VLC, Vedantu Learning Center in JP Nagar, and we have a lot of students there. So, I would request all of you to make a call on this number and just check when can you book a slot. So slot book ಮಾಡಿ ನಿಮ್ಮ ಪೇರೆಂಟ್ಸ್ ಜೊತೆ ಅಲ್ಲಿ ಹೋಗಿ just look at the entire center, ಯಾವ facilities ಇದೆ, ಯಾವ books refer ಮಾಡ್ಬೋದು, what are the teachers there, how are the teachers there. We will also be coming there once in a while to give you mentorship. So if you are a student who is starting, who doesn't know anything and wants to crack NEET in 2027, 2026, then you can join the Vedantu Learning Center if you are a student who is looking for offline. ಆನ್ಲೈನ್ ಬೇಕಂದ್ರೆ ನಮ್ಮ ಪ್ರೋ ಬ್ಯಾಚಸ್ ಇದೆ, ಅದು ಡಿಸ್ಕ್ರಿಪ್ಷನಲ್ಲಿ ಲಿಂಕ್ ಇರುತ್ತೆ, ಸೊ ಅದನ್ನು ಕ್ಲಿಕ್ ಮಾಡ್ಬೋದು. ಓಕೆನಾ? ಎಸ್, ಇಟ್ ವಿಲ್ ರಿಜೆಕ್ಟ್ ಇಟ್ಸ್ ಓನ್ ಪೋಲನ್. ಓಕೆ, ಜಸ್ಟ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ಜಸ್ಟ್ ಗಿವ್ ಮಿ ಅ 5 minutes ಬ್ರೇಕ್. ಓಕೆ, ಐ ವಿಲ್ ಬಿ ಬ್ಯಾಕ್. ಡೋಂಟ್ ಗೋ ಎನಿವೇರ್. ಕ್ವಿಕ್ಲಿ ಮಾರ್ಕ್ ದಿ ಅಟೆಂಡೆನ್ಸ್. ಓಕೆ, ಎಸ್ ಅಮಿತ್. Okay, let's continue. Come on. Viability done. Next topic is pole and pistil interaction. Okay. Pole and pistil interaction andre students. Pollen matte stigma, they go through a conversation. Okay, like a conversation means if this pollen is the right one for me or if it is the wrong one for me. Just like in our lives also, we meet a lot of people. Some people are the right people for us, some people are the wrong people for us. How do we know this? After we talk to them for a while, after we sit with them and chit chat for a while, maybe it takes 1 month, 2 months, 3 months, 1 year, 3 years. It might even take an entire lifetime to understand if this person is right or wrong. Aatarane, pollen grains, many pollen grains come and land on the stigma. But stigma has to decide which is the one that is compatible for it and which is the right choice for it. So, adikon pollen pistil interaction martare. First one is called as exchange of chemical dialogues. Chemical dialogues andre, they are going to, stigma is going to release a chemical for the pollen grains to start its germination. Okay. Then you have pollen tube germination. Pollen tube moving towards the egg. As you can see, this two pollen got rejected. This pollen continued to move all the way towards the egg. Why? Because the pollen tube olagade enide male gamete ide. Ee male gamete ellog fuse agbeku, egg jote fuse agbeku alva. So style inda move agi ovary, ovule, embryo sac, cytoplasm of the synergist matte egg. This is how the entire formation continues. Okay, clear to all of you? So students, in our life also, when we, let's say when we want to choose a partner, okay, we might get one doctor, engineer, businessman, one person who doesn't have a proper job also. Now depending on what we want, we will reject or accept them. After accepting them, we will let them talk to our family and continue the process. Atarane illu. Okay, atarane same way here also. So look here, here you can see male gamete. Okay, this is the NCERT diagram. Male gamete, vegetative nucleus. Okay, vegetative nucleus is there, male gamete is there, filiform apparatus. So embryo sac alli kelagade synergist ittu, male antipodal cell, central polar nuclei ittu. A synergist hatra filiform apparatus ittu, a finger like structures which are going to help the guiding of the pollen tube. Okay, which will allow the guiding of the pollen tube. Now the pollen tube will go where? Towards the egg it is going to go. Why? It has to fertilize the egg. Clear to all of you? Now, students, normally 2 cell stage alli, one vegetative cell, one male gamete andre, that male gamete is going towards the egg. But third 3 cell stage andre, enagutte, one vegetative cell matte 2 male gametes iruttallva, onde egg irodu, 2 male gametes idre problem agutte. So innond male gamete will go and fuse with the polar nuclei which is right at the center, So male gamete will fuse with the egg, it will form zygote. Male gamete matte egg ಎರಡು haploid ಆಗಿರುತ್ತೆ, NN. It will form a zygote which is diploid. Okay, this formation is called as syngamy. Okay, ಇಲ್ಲಿ ನೋಡಿ ಇನ್ನೊಂದು male gamete ಇತ್ತಲ್ವಾ, 3 cell stage ಇದು. ಇನ್ನೊಂದು male gamete polar nuclei ಜೊತೆ fuse ಆಗಿ it will form primary endosperm cell or PEC which is 3N. ನಿಮ್ಮ NCERT ಅಲ್ಲಿ ಪೆನ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ, ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಬರೀಬಹುದು, ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ಸೆಲ್ ಅಂತಾನು ಬರೀಬಹುದು. ದಟ್ ಇಸ್ ಗೋನ್ ಬಿ 3N. ಅಂದ್ರೆ ಮೇಲ್ ಗ್ಯಾಮಿಟ್ ಪ್ಲಸ್ ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಇಸ್ ಗೋನ್ ಬಿ 2N, ಮೇಲ್ ಗ್ಯಾಮಿಟ್ ಇಸ್ ಗೋನ್ ಬಿ N, ಫ್ಯೂಸ್ ಆಗಿ 3N ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ ವಿಚ್ ಇಸ್ ಪ್ರೈಮರಿ ಎಂಡೋಸ್ಪರ್ಮ್ ನ್ಯೂಕ್ಲಿಯಸ್. ಓಕೆ, ಅದನ್ನ ನಾವೇನ್ ಕರಿತೀವಿ? ಟ್ರಿಪಲ್ ಫ್ಯೂಷನ್. ಸೊ 2 cell stage alli syngamy nadiyutte, but 3 cell stage alli syngamy matte triple fusion ottige nadiyutte. So we call it double fertilization. Syngamy plus triple fusion, double fertilization. Clear? So here remember, syngamy will end up forming zygote. Triple fusion will give us primary endosperm cell that is going to ultimately form the endosperm. Now what is endosperm? ಎಂಬ್ರಿಯೋ ಗ್ರೋ ಆಗುತ್ತಲ್ವಾ, ಝೈಗೋಟ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗೋ ಟೈಮಲ್ಲಿ ಅವ್ರಿಗೂ ಎಲ್ಲ ನ್ಯೂಟ್ರಿಷನ್ ಪ್ರೊವೈಡ್ ಮಾಡಬೇಕು. ಸೊ ಅದಕ್ಕೆ ನಾವು ಅದನ್ನು ಎಂಡೋಸ್ಪರ್ಮ್, ಓಕೆ, ಎಂಡೋಸ್ಪರ್ಮ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತೀವಿ. ಸೊ ದ್ಯಾಟ್ ಮೋರ್ ಫುಡ್ ರಿಸರ್ವ್ ವಿಲ್ ಬಿ ದೇರ್, ಪ್ರೊವೈಡ್ಸ್ ನರಿಶ್ಮೆಂಟ್ ಟು ದ ಗ್ರೋಯಿಂಗ್ ಎಂಬ್ರಿಯೋ. ಕ್ಲಿಯರ್ ಟು ಆಲ್ ಆಫ್ ಯು? Clear to all of you? Yes or no? Done? So at every single stage there is some person to provide nourishment. Okay, now artificial hybridization. Artificial hybridization andre, now let's say I want to, produce something that is of a hybrid quality, that is high yielding. So adik nan en martini, I will not let the plant decide how to do pollination. I will only decide how. This is a flower. From this flower, I am going to remove both the anthers. Why? So that self-pollination can be avoided. Anther idre, pollen grains will fall on the stigma, self-pollination kandita agutte. Adanna avoid madbekandre, nan en madbeko, anther na remove madbeko. Our process again, ನಾವು ಏನು ಕರಿತೀವಿ ಅದನ್ನ emasculation. Okay, making it a less of a masculine. ಆ ಥರ ಹೇಳ್ಬೋದು, ನಾರ್ಮಲ್ ಲ್ಯಾಂಗ್ವೇಜ್ ಅಲ್ಲಿ ಆ ಥರ ವರ್ಡ್ಸ್ ಯೂಸ್ ಮಾಡ್ತಾರೆ. Emasculation ಅಂದ್ರೆ removal of anther from the flower with the help of forceps. So now anther will get removed. So students, obviously anther remove ಮಾಡಿದ್ರೆ ಪ್ರಾಬ್ಲಮ್ಸ್ ಎಲ್ಲ ಕಮ್ಮಿ ಆಗಲ್ಲ, ಬೇರೆ pollen grains ಬಂದು ಬೀಳುತ್ತಲ್ವಾ. We have to avoid that also. ಅದನ್ನ ಅವಾಯ್ಡ್ ಮಾಡೋಕ್ಕೆ ಒಂದು ಪಾಲಿಥಿನ್ ಬ್ಯಾಗ್ ಇಲ್ಲ ಅಂದರೆ ಒಂದು ಬಟರ್ ಪೇಪರಲ್ಲಿ ನಾವು ಆ ಫ್ಲವರ್ನ ಕವರ್ ಮಾಡ್ತೀವಿ, ವಿಚ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಬ್ಯಾಗಿಂಗ್. ಸೊ ಆರ್ಟಿಫಿಷಿಯಲ್ ಹೈಬ್ರಿಡೈಸೇಷನ್ ಹ್ಯಾಪನ್ಸ್ ಇನ್ 2 steps, emasculation and bagging. Emasculation, we remove the anther with the help of forceps. Bagging ಅಂದರೆ we use a paper cup, not paper cup, ಬಟರ್ ಪೇಪರ್. ಓಕೆ, ಸಡನ್ಲಿ ನನ್ನ ಬ್ರೈನ್ ಗ್ಲಿಚ್ ಆಯಿತು. ಈ ಬರ್ಫಿಂಗ್ ಇದು ತೋರಿಸೋದಲ್ಲ ಇಂಟರ್ನೆಟ್ ಅಂದರೆ ಬಫರಿಂಗ್, ಸೊ ಅದು ಹೋಯ್ತು. ಓಕೆ, ಬಫರಿಂಗ್ ಆ ಥರ ನನ್ನ ಬ್ರೈನು ಅಂತ 2 secondsಗೆ ಬಫರ್ ಆಯ್ತಾಯಿತ್ತು. ಓಕೆ, ಸೊ ಹಿಯರ್ ದಿಸ್ ಹ್ಯಾಪನ್ಸ್ ವೆರಿ ಕಾಮನ್. ಓಕೆ, ಯು ಶುಡ್ ಸಿ ಮೆನಿ when we teachers all sit together and we talk, this is what we tell. Suddenly brain just stops, giving all the inputs, because we have to remember all of this, so suddenly brain will be like, no, I don't want to do anything, so it will just stop. So our situation is. So here nodi bagging, and then it will cover the entire flower has to protect from any other unwanted pollen grains, covering the emasculated flower with the butter paper. Now after that, I am the one who is deciding, so I will dust the pollen grains of the desired and I will again cover it with a bag. Why? So that to avoid any kind of contamination. So desired pollen grains are dusted, rebagging is done. So artificial hybridization, ee tara diagrams draw ಮಾಡಿ ಯಾರು ನಿಮ್ಮನ್ನ ಓದ್ಸಲ್ಲ, so that's why study now. Artificial hybridization, emasculation, where you remove the anther. Bagging, where you cover it with butter paper. Then you add, dust the desired pollen and then you do rebagging. Clear? Clear. Done. Okay, now we will start with post-fertilization events. Post-fertilization event andre, ovule will form the seed, ovary will form the fruit. Okay, ovary will form the fruit. Now look here, the cells of this tissue are filled with reserve food materials. ಯಾರ ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ? ಎಂಡೋಸ್ಪರ್ಮ್ ಬಗ್ಗೆ ಮಾತಾಡ್ತಾ ಇದ್ದಾರೆ, because endosperm is the one who will provide the food reserve material. Pen undergoes successive nuclear division to give rise to free nuclei. Free nuclear endosperm becomes cellular. So I'll teach you this. Okay, if you read this you will not understand. Look here, this is your endosperm, this is the pen, primary endosperm nucleus, this is the pec, primary endosperm cell. This will undergo karyokinesis. Karyokinesis andre eno, nucleus divide agadu. Okay, so ಇಲ್ಲಿ nucleus divide ಆಗಿದೆ, zygote ಇಲ್ಲಿರುತ್ತೆ. Okay, karyokinesis will continue, no cytokinesis. So karyokinesis ಈ ತರ ಕಂಟಿನ್ಯೂ ಆಗ್ತಾ ಇರುತ್ತೆ, ನ್ಯೂಕ್ಲಿಯಸ್ ಇನ್ಕ್ರೀಸ್ ಆಗ್ತಾನೆ ಇರುತ್ತೆ. ನಂಬರ್ ನೋಡಿ, ಇಲ್ಲಿ 4 ಇತ್ತು, 4 ಇಂದ 8 ಫಾರ್ಮೇಷನ್ ಆಗಿತ್ತು. ಸೊ ಇಲ್ಲೆಲ್ಲ ಸೈಟೋಕೈನೆಸಿಸ್ ಆಗ್ತಾನೆ ಇಲ್ಲ, ಸೆಲ್ಯುಲರ್ ಫಾರ್ಮೇಷನ್ ಎಲ್ಲೂ ಆಗ್ತಾ ಇಲ್ಲ, ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನು ಎಲ್ಲೂ ಆಗ್ತಾ ಇಲ್ಲ. Okay, so example, tender coconut water. The tender coconut water that you're drinking is a, what is that? It is a nuclear division. Okay, nuclear division. After some time, students, what is going to happen? Cytokinesis will happen. Andre, cell wall formation will happen. And nodi, ella nucleus, around every nucleus, there will be cell wall formation leading to cellular endosperm. The white kernel ಏನು ಹೇಳ್ತೀವಿ ಕನ್ನಡದಲ್ಲಿ ನನಗೆ ಗೊತ್ತಿಲ್ಲ ಕರೆಕ್ಟಾಗಿ, ಎಳ್ನೀರು, ಅದ್ರಲ್ಲಿ ನೀರು ಇರುತ್ತಲ್ವಾ, ನೀರು ಬಂದ್ಬಿಟ್ಟು ಫ್ರೀ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನು. ಆ ವೈಟ್ ಕಲರ್ ನಾವು ತಿಂತೀವಿ ಅಲ್ವಾ, ಅದು ಬಂದ್ಬಿಟ್ಟು ಸೆಲ್ಯುಲರ್ ಡಿವಿಷನ್. ಓಕೆ, ಸೊ ನ್ಯೂಕ್ಲಿಯರ್ ಡಿವಿಷನ್ ಹೆಂಗಾಗೋದು? ಕಂಟಿನ್ಯೂಸ್ ಕ್ಯಾರಿಯೋಕೈನೆಸಿಸ್ ಆಗುತ್ತೆ, ಸೈಟೋಕೈನೆಸಿಸ್ ಆಗಲ್ಲ. ಅದಾದಮೇಲೆ ಸೈಟೋಕೈನೆಸಿಸ್ ಆದರೆ ಸೆಲ್ ವಾಲ್ ಫಾರ್ಮೇಷನ್ ಆಗುತ್ತೆ, ದಟ್ ಇಸ್ ಗೋನ್ ಬಿ ಯುವರ್ ಸೆಲ್ಯುಲರ್ ಎಂಡೋಸ್ಪರ್ಮ್. ಕ್ಲಿಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್? ಅಟ್ ಲೀಸ್ಟ್ ಟೆಲ್ ಇಫ್ ಯು ಅಂಡರ್ಸ್ಟುಡ್ ಆರ್ ನಾಟ್, ವಾಟ್ ಇಸ್ ದಿಸ್ ಕೈಂಡ್ ಆಫ್ ರೆಸ್ಪಾನ್ಸ್? You only come here to tell hi to your friends. This Karnataka channel is made so you can make a WhatsApp community. Tell at least you understood or not. You're sitting in the class ideally. ಇಮ್ಯಾಜಿನ್ ಮಾಡಿ ನಾನು ಕ್ಯಾಮ್ರಾ ನೋಡಿ ಎಷ್ಟು ಟೈಮ್ ಮಾತಾಡ್ತೀನಿ, ನಿಮ್ಮ ರೆಸ್ಪಾನ್ಸ್ ನನಗೆ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್. ಶೇರ್ ಮಾಡೋಕ್ಕೆ ಹೇಳಿದ್ರೆ ಅದು ಮಾಡಲ್ಲ, ಲೈಕ್ ಮಾಡೋಕ್ಕೆ ಹೇಳಿದ್ರೆ ಅದು ಮಾಡಲ್ಲ, ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡೋಕ್ಕೆ ಹೇಳಿದ್ರೆ ಅದು ಮಾಡಲ್ಲ, ಬಟ್ ಯು ಪೀಪಲ್ ವಾಂಟ್ ಆಲ್ ದ ಕಂಟೆಂಟ್. Students, we are NEET teachers. Okay, we have been training students for NEET from past 2 years. We are teaching you biology for PUC and for your KCET. So what more do you want? We are going to build you such a good foundation. So I want all of you to be more active, give more response. It is very difficult to stand like this and teach. I was an offline teacher before. Okay, so it is very difficult when I stand like this and no one is responding. At least tell yes ma'am, at least put a thumbs up. Okay, be active in the class. Otherwise, you know, our hope for the Karnataka students will go down. Understood? Okay. Yes, thank you, Kishan. Okay, now look here. Here, cytokinesis, that means cell wall formation, developing embryo will be there. Endosperm 3N, nourishment for the developing embryo. Zygote will slowly start forming embryo. Clear? Now, whatever is mentioned in your NCERT, I have drawn and taught that. Okay? So, ill nodi students. Zygote ಹೆಂಗೆ ಡೆವಲಪ್ ಆಗುತ್ತೆ, zygote will form pro-embryo, okay. Then from there we will form globular embryo. From globular embryo, heart-shaped embryo will ultimately form a mature embryo which will have a plumule, radicle and a cotyledon. Plumule is the one that is going to form the shoot, radicle is the one that will form the root, R for radicle, R for root. Cotyledons are the food reserve materials, okay. So, ನಾರ್ಮಲ್ ಆಗಿ ಝೈಗೋಟ್ ಇತ್ತು, ಅದು ಪ್ರೋ ಎಂಬ್ರಿಯೋ ಆಯಿತು, ಪ್ರೋ ಎಂಬ್ರಿಯೋದಿಂದ ಗ್ಲೋಬ್ಯುಲರ್ ಎಂಬ್ರಿಯೋ, ಬಟ್ ದೀಸ್ ಆರ್ ಸಸ್ಪೆನ್ಸರ್ಸ್, ಓಕೆ. ಹಾರ್ಟ್ ಶೇಪ್ಡ್ ಎಂಬ್ರಿಯೋ, ಮೆಚೋರ್ ಎಂಬ್ರಿಯೋ. If you don't remember that much also, it's fine. Mature embryo is important for you, okay. Mature embryo is important for you. See this one, a diagrammatic representative of mature dicot embryo. ಇಲ್ಲಿ ನೋಡಿ ಏನೆಲ್ಲ ಇದೆ, plumule which will become the shoot, cotyledons are there, 2 cotyledons are there, hypocotyl is there, radicle is there, root cap is there. Root cap, why students? So that the root will be going through harsh, you know, grounds, okay, where there will be water, where there will be no water. So adanna protect madake root cap is there, okay. Root cap will be given to them. Now, okay, this you understood, plumule. This will be asked. Types of seeds, okay, types of seeds will be asked for you. Types of seeds means one which is called as non-albuminous seed, one which is called as albuminous seed. Non-albuminous seed andre no endosperm, seed utilized entire endosperm. Endosperm eno food reserve alva. Endosperm was the food reserve. If the entire food reserve is utilized, that kind of seed we call as non-albuminous seed, okay. Non-albuminous seed. Remember PUBG game ellargu gotala, P for P, U enu illa, B for beans, G for groundnut. Example. So endosperm was there for the embryo to utilize. Full endosperm they utilized of. All the seeds utilized full endosperm. Adik now non-albuminous seeds anda karitivi. Illi nodi, endosperm was there. Some endosperm is going to remain. Okay, seed does not utilize entire endosperm. Endosperm retains. This is called as what? Albuminous seed. Example, castor, barley, wheat. Okay, castor, barley and wheat is the example. Can you remember this much? So here they have given, see, the embryo are simple structures, generally thick and swollen due to the storage of food reserves. Mature seeds may be non-albuminous or exalbuminous. Non-albuminous seed have no residual endosperm. ಎಷ್ಟು ಎಂಡೋಸ್ಪರ್ಮ್ ಕೊಟ್ಟಿದ್ದೀವಿ ಅದು ಫುಲ್ ಅವ್ರು ಯೂಸ್ ಮಾಡ್ತಾರೆ, ಓಕೆ. ನೋ ರೆಸಿಡ್ಯೂಯಲ್ ಎಂಡೋಸ್ಪರ್ಮ್ ಆ್ಯಂಡ್ ಇಟ್ ಇಸ್ ಕಂಪ್ಲೀಟ್ಲಿ ಕನ್ಸ್ಯೂಮ್ ಡ್ಯೂರಿಂಗ್ ದ ಎಂಬ್ರಿಯೋ ಡೆವಲಪ್ಮೆಂಟ್. ಎಕ್ಸಾಂಪಲ್ ಪಿ, ಗ್ರೌಂಡ್ನಟ್, ಓಕೆ. ಎಕ್ಸಾಂಪಲ್ albuminous seed ಅಂದರೆ they retain a part of the endosperm as it is not completely used during the embryo development. Example, wheat, maize, barley, castor. Wheat, maize, barley, castor. Occasionally in some seeds there will be persistent nucellus. Ma'am, nucellus, ma'am, I forgot. Nucellus, all of you remember? When we were talking about megaspore mother cell, there were nucellus cells. That nucellus, one of them was like, I want to be the megaspore mother cell, that oversmart nucellus. That is going to be persistent. Nucellus usually full consume ಆಗುತ್ತೆ, embryo sac formation ಆದ ಮುಂಚೆನೇ ಎಲ್ಲ consume ಆಗುತ್ತೆ. But sometimes, in some scenarios, this nucellus is going to be persistent. Persistent ಅಂದರೆ, I am not going to go anywhere, you go wherever you want, I will stay with you, okay. That persistent nucellus is going to be called perisperm. Okay, such such nucellus is called as perisperm. Example banbit BB house, remember my students. Big Boss house ilva? That is black pepper and beet. Okay, Big Boss house anta remember madi. So now I have given you PUBG and BB house to remember. Okay, BB house to remember. Yes, now okay, see, this is what I told you. Both the integuments illa, outer matte inter integuments, they are going to form the seed coat, testa and tegument. Okay, testa and tegument. So every one ultimately has a job. Ovule alli integuments ittu, hilum ittu, micropylar end ittu, everyone has a job. What is the beat? Okay, now, okay, fertilized ovule. Students, ovule which is fertilized is ultimately called the seed. Fertilized ovary is called the fruit. Fertilized ovule is called the seed. Okay, so that means fruit matte seed formation simultaneously agutte. First fruit tinbit, then seed will be formed. No, both will be happening together. Now, integument, seed coat, wall of the ovary, fruit. Wall of the ovary, no, ovary wall will be there, right? That ovary wall will form the pericarp. Ovary wall will form the pericarp. This you would have studied in morphology of flowering plants, class 11th alle. Okay, so pericarp formation will happen. You'll notice, students, the microbial remains as a small pore in the seed coat. This facilitates the entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and the seed relatively dries. Okay, that is 10 to 15% moisture by mass. The general metabolic activity of embryo slows down. The embryo may enter a state of something called as dormancy. It will enter what? Dormancy. Students, night time when you are sleeping, you go to a state of dormancy and complete inactiveness. Brain work agala, body work agala, yen bageno think madala, maybe some dreams and all will come, but adre tension iralla. But ee working time I have to remember so much content, I have to write this test, this girl didn't talk to me, this boy didn't talk to me, idella full continuous thought process nadithalva. So seed what they will do when conditions become unfavorable. Okay, when conditions are not favorable, avuru relatively dry agutte. Because water idre seed will become bad. So adike they will relatively remove all the water and they will become dry. When condition again becomes favorable, they will start germinating slowly. Adike neevu normally nodirtira when your students in some rock and all, okay, some rock, let's say some rock like this, okay, one seed has come and fallen, okay, one seed has come and fallen. But you will notice that the seed, you will never notice that seed. Why? Because the seed has gone to a stage of dormancy. Okay, seed has gone to a stage of dormancy and it is relatively, okay, it is relatively dry. That means 10 to 15% it has completely removed the water. Okay, no water content. Why? Because water content is there, they are going to become bad. After some time, let's say condition becomes favorable, okay, it starts raining, okay, it starts raining and it is, getting enough sunlight and all of it, okay, everything is there, okay, okay, sun is there, everything is there, condition has become favorable, slowly ನೀವೇನು ನೋಡ್ತೀರಾ ಈ seed ಇಲ್ಲಿಂದ germinate ಆಗಕ್ಕೆ ಶುರು ಮಾಡ್ತಾರೆ, ಈ ತರ germinate ಮಾಡಕ್ಕೆ ಶುರು ಮಾಡ್ತಾರೆ. Understood? Yes or no? Okay, they start germination. So this is how you should decode your NCERT lines. Okay, confuse agvedi, you ma'am NCERT artha agta illa. Or in favorable conditions are available, that means adequate moisture, oxygen, suitable temperature ella idre, they will germinate. As the ovules mature into seed, the ovary develops into fruit. The transformation of ovules into seed and ovary into fruit proceeds simultaneously, continuous ಆಗಿ ಆಗ್ತಾ ಇರುತ್ತೆ. First PU exam, second PU exam continuous ಆಗಿ ನಡೀತಾ ಇಲ್ವಾ? First PU exam ಒಂದು ರೂಮಲ್ಲಿ ನಡೀತೆ, second PU exam ಇನ್ನೊಂದು ರೂಮಲ್ಲಿ ನಡೀತೆ. Separate ಅಲ್ಲ. So the walls of the ovary develop into the wall of the fruit called pericarp. Okay, they develop into what? Pericarp. The fruits may be fleshy as in guava, orange and mango, and it might be like different as in coconut and all. Okay, now fruits are, students, two types, true fruits matte false fruit. Okay, true fruits andre the ovary, so generally ನಾವು ಏನು ಹೇಳ್ತಾ ಇದೀವಿ, ovary fruit formation ಮಾಡಬೇಕು, right? Ovary will form the fruit. So if ovary is forming the fruit, I will call it true fruit. If ovary, other than the ovary, any other part is forming the fruit, I will call it false fruit. So other parts excluding the ovary forms the fruit, like thalamus. Example, apple. Apple ನೀವು ತಿಂತಿರಲ್ವಾ, what you are eating is the thalamus. What you are eating is the thalamus. Okay. So that means apple is a false fruit. Okay. ಇಲ್ಲಿ ನೋಡಿ, fleshy fruit, again mesocarp, endocarp, pericarp. This again not required for you now. Okay. False fruit, true fruits you understood. Partheno. Fruits develop without fertilization. You'll be like, huh? Full chapter you taught and now you're telling me Rama doesn't exist. So like that only this is fruit without fertilization. So fruits can form without fertilization. That condition is called parthenocarpic fruit, and banana is one such example. Without fertilization fruit form agutte, that is called as parthenocarpic fruit. Banana is one such example. Parthenocarpy can be induced through application of growth hormones. Okay, and such fruits are seedless. Banana doesn't have seed, no, even though that black spot we tell. Okay, clear? Okay, now, okay, then Asteraceae and grasses have evolved special mechanism to produce seeds without fertilization. So first now fruit without fertilization, okay, which is called parthenocarpy. Now we are studying seed without fertilization, which is called as apomixis. Okay, idella definition are kelboth students, 2 mark ige apomixis. So what is fruit production without fertilization called? Parthenocarpy. Apomixis is an asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, diploid egg cell, okay, diploid egg cell is formed without reduction. Andre normally diploid cell N in the form agutte, but ille en agutte MMC directly egg form madre, it is going to go through 2N. So this egg will be like, okay, I do not want any male or anything, I will only form the seed. So reduction without going through reduction division, and it will directly develop into embryo. This is one scenario of apomixis. Okay, apomixis scenario 1. Second scenario, this is going to happen in citrus and mango. Okay, citrus and mango. Second scenario ಏನಂದ್ರೆ students, the nucilla cells ilva, nucilla cells will directly start surrounding the embryo sac, and they will start dividing, protruding into the embryo sac and developing into embryos. So this nucilla cells, one by one, all of them will start forming embryos. Okay, many embryos. So in such species, each ovule contains many embryos. Occurrence of more than one embryo in the seed will lead to polyembryony, which you see in citrus and mango. Citrus and mango. So polyembryony, apomixis, parthenocarpy. More condition odbeko. Okay na? Understood? Chapter is over. Chapter is over. Did you all understand the whole chapter? Now next I will be doing questions from this chapter. Not now, later. Okay, so I want all of you to go to the channel. Okay, so this is the channel that you are watching. Okay, so go here, click on this. Okay, if you go here, you can see physics videos have already come on the channel. Electric charges, okay, FA1 videos have come, and those who are looking for KCET option entry, il videos already there. KCET ಬಗ್ಗೆ ತುಂಬಾ videos ಮಾಡಿದ್ದೀನಿ. Counseling mistakes avoid ಮಾಡಿ. This is for NEET students. Okay, I have also done about the cutoff and option entry. Okay, this one, what to do if your KCET rank is low, adu ಮಾಡಿದ್ದೀನಿ. So ಇವಾಗ ನೋಡಿ FA1 class continue ಆಗ್ತಾ ಇದೆ. First PUC class, KCET class ನಡೀತಾ ಇದೆ. ಇದೆಲ್ಲ first chapters ಇಂದ, so that you people can study. Okay, reproductive health complete ಮಾಡಿದ್ದೀನಿ. So make sure that you are studying. Okay, motion in straight line happening continuously. So see, all the videos are already on the channel. Okay, molecular basis of inheritance also done, biological classification done. So make sure that you are studying. Every day classes are happening on the channel. Okay, what you have to do is you have to come to the channel, study from the channel, and subscribe to the channel. Okay, yes, we have to make it 40K very soon, so I want all of you to subscribe to the channel. Okay, so students, sexual reproduction, flowering plants, impossible task. 1 hour 45 minutes ಅಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದೀನಿ. ಸೊ ಇದು ಬಂದ್ಬಿಟ್ಟು ಕ್ವಿಕ್ ರಿವಿಷನ್. ಡೀಟೇಲ್ ಲೆಕ್ಚರ್ ಆಲ್ರೆಡಿ ಚಾನಲಲ್ಲಿ ಇದೆ. ಬಟ್ ದಿಸ್ ಇಸ್ ಫಾರ್ ಆಲ್ ದ ಸ್ಟೂಡೆಂಟ್ಸ್ ಆರ್ ಪ್ರಿಪೇರಿಂಗ್ ಫಾರ್ ಫಸ್ಟ್ ಯೂನಿಟ್ ಟೆಸ್ಟ್. ಲಾಸ್ಟ್ ಮಿನಿಟ್ ಒಂದು ರಿವಿಷನ್ ಮಾಡಬೇಕಂದ್ರೆ ಈ ಒಂದು ಲೆಕ್ಚರ್ ಸಾಕು, ಬೇರೆ ಏನೂ ಮಾಡೋಕ್ಕಿಲ್ಲ. Just watch this lecture. I can guarantee you that you're going to write your exam very well. ಈ lecture ನೋಡಿ ಅದಾದಮೇಲೆ ನಾನು ಕ್ವೆಶನ್ ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡ್ತೀನಿ. 2 marks 3 marks 5 marks ಕ್ವೆಶನ್ ಬ್ಯಾಂಕಿಂದ ಸಾಲ್ವ್ ಮಾಡ್ತೀನಿ, ನಾಟ್ ಸಮ್ ರ್ಯಾಂಡಮ್ ಪ್ಲೇಸ್. ಸೊ ಆ ಕ್ವೆಶನ್ ಬ್ಯಾಂಕಿಂದ ಸಾಲ್ವ್ ಮಾಡೋ ಕ್ವೆಶನ್ಸು ಪ್ರಾಕ್ಟೀಸ್ ಮಾಡಿ. ಫಸ್ಟ್ ಯೂನಿಟ್ ಟೆಸ್ಟ್ like this you will get full marks. Okay, so yes, thank you so much for joining. I hope all of you enjoyed the class. Please let me know in the comments how the class was, and do not forget to smash the like button and subscribe to the channel. These are the small few rewards that we ask you for, so that we can grow the channel bigger and we can give you so much content. Okay, because if the channel is small, content is limited, we cannot bring in more good teachers. So if you want to bring in more good teachers, I want all of you to be cooperative, because Karnataka students deserve having a proper channel that helps them in PUC, KCET, NEET, Comet K, whatever exams they're preparing. We want to help you. Okay, so thank you so much, students. See you all in next class. Till then, take care. Bye bye.